Hi everyone, welcome to my best ever all-in-one Edexcel IGCSE Biology video. I am so excited to share this with you. This is 4 hours of biology perfection as far as I'm concerned. I'm going to take you through in just the right amount of detail you'll need for your exam, as well as showing you past paper questions to help you perfect your exam technique to make your dream grade a totally achievable goal. I hope you see by the time you've sat through this 4 hours that this has been time extremely well spent. I'm super proud of this video and I hope you enjoy it. Just to introduce myself, my name's Hazel, I'm Science with Hazel. I studied at St John's College, the University of Cambridge. I'm a qualified secondary science teacher and I've been working as a private tutor for over 10 years, so I think I'm well placed to make this content. Throughout you'll see snippets of my revision guide. If you'd like to get hold of a copy, that's available on my website, SwH Learning. There's information there about private tuition if you want help from me or any of our tutors, as well as if you want to sign up to our online revision courses which are held on Zoom that will give you the exam preparation you need to fully ace your exams. But let's just get going. Now, to begin with, what are the characteristics that all living organisms share? Well, we're going to use Mrs. H. Gran to help us remember. The M stands for movement, R stands for respiration, S stands for sensitivity, H stands for homeostasis, G is growth, R is reproduction, E is excretion, and N is nutrition. As you'll notice throughout my videos, I'm going to be really concise here. I'm not going to talk about all the extra stuff that you might have been taught. I just want to teach you the things which will most likely come up in the exam. So the R, which stands for respiration, do make sure you can write the word equation and the balanced symbol equation here. You've got oxygen plus glucose produces carbon dioxide and water and releases energy. Make sure you never say produces energy, you won't get any marks for that. It releases energy. In terms of the balanced symbol equation, we have 6O2 reacting with C6H12O6 goes to 6CO2 plus 6H2O. Now, sensitivity refers to an organism's ability to respond to its surroundings. H, homeostasis, this is the maintenance of a steady internal environment. That's important when we consider mammals. They need to keep their body temperature around 37 degrees. That's to ensure that the enzymes have the optimum temperature they need to work. We need to control our blood glucose levels as well as our blood water levels in a process known as osmoregulation. These are all examples of homeostasis. Reproduction is all about increasing the number of organisms. E, excretion, this is the removal of metabolic waste. What kind of substances do we excrete? Well, we excrete sweat from our skin, carbon dioxide from our lungs, urea from our kidneys. N stands for nutrition, and that's all to do with feeding. The variety of living organisms. So we're going to divide various types of organism into whether they are prokaryotes or eukaryotes, and you do need to be able to distinguish between these two terms. So fundamentally speaking, the term eukaryote refers to the fact that an organism contains membrane-bound organelles. This means that the subcompartment of cells have membranes around them. So that could include something like a nucleus, a chloroplast, a mitochondria. These are all examples of membrane-bound organelles. So examples of eukaryotes include plant cells, fungi cells, animal cells, whereas prokaryotes do not contain membrane-bound organelles, so they don't contain a nucleus, chloroplast, mitochondria. A good example of prokaryotes are bacterial cells. Remember, rather than having a distinct nucleus, bacterial cells have a circular chromosomes known as a nucleoid, as well as small circles of DNA known as plasmids. Let's go into greater detail now about the components of an animal cell, plant cell, etc. So an animal cell contains a nucleus. This controls the activities of the cell. Please do not write that it is the brain of the cell in the exam. You won't get any marks for that. The mitochondria is the site of aerobic respiration. This is where energy is released. Do make sure you can state the equation of glucose plus oxygen goes to carbon dioxide plus water plus energy. The cell membrane controls the entry and exit of substances into and out of the cell. The cytoplasm is where chemical reactions occur, and the ribosomes are very important because they are the site of protein synthesis. This is where proteins are made. You'll find out lots about ribosomes in the transcription and translation stages of protein synthesis. Plant cells now. Plant cells contain the same organelles as animal cells, so if you've learned the functions of the cytoplasm and the nucleus, etc., that's still relevant here. But in addition, plant cells contain 3 extra organelles. Those are the chloroplasts. This is where photosynthesis occurs. Photosynthesis is when you take carbon dioxide and water, they react together to produce glucose and oxygen. This reaction requires light energy, and it's the way that plants can make their own food. Plant cells also contain a vacuole, which is filled with cell sap, and they have a cellulose cell wall, which helps to protect and maintain the shape of the cell. Looking closely now at the bacterial cell, which remember we said is prokaryotic. I've already told you they don't contain a distinct nucleus. Instead, you'll find a circular chromosome known as a nucleoid, and small circular strands of DNA called plasmids, which can be used in genetic engineering to produce substances like insulin. Bacterial cells also have cytoplasm, they have a cell membrane, they have a cell wall. They may also have a small tail known as a flagellum, which helps the bacterial cell to move. Now, a fungi cell. Examples of fungi include yeast. Fungi cells have a cell wall made out of chitin, they have a cell membrane, a nucleus, cytoplasm, mitochondria, and ribosomes. They could ask you to distinguish between how the various cells store their carbohydrate. So plant cells store their carbohydrate as starch, whereas animal and fungi cells store their carbohydrate as glycogen. You'll find out differences about the structure of both starch and glycogen if you study A-level or IB biology. In terms of extra differences, bacteria are unicellular, which means they're made up of one cell. You can have some bacterial cells which are pathogenic. This refers to the fact that they are disease-causing. Other bacterial cells are harmless. Key examples include pneumococcus, which causes the disease pneumonia, and lactobacillus bulgaricus, which can be used to make yogurt from milk. Fungi cells can be either unicellular or multicellular. Yeast is a good example of a unicellular fungi. Mucopol is an example of a multicellular fungi. In multicellular fungi, their structure is incredibly specialized in that they are made up of strands known as hyphae, which join together to form a network known as mycelium. Fungi cells carry out saprotrophic nutrition. This means that they excrete enzymes extracellularly, that means outside of their bodies, onto dead matter such as leaves in woods. And then these enzymes digest that dead leaf matter so that they can then be absorbed by the fungi cells. The Protoctis, we call these the dustbin kingdom because sometimes they can be animal-like, such as an amoeba, sometimes they can be plant-like, such as Chlorella. They can be either unicellular or multicellular. One Protoctis to be aware of is Plasmodium, which is responsible for the disease malaria. Remember, the mosquito does not cause malaria, it acts as a vector. It transports the Plasmodium to the host, the human. The virus cells now, these are non-living, and this is because they do not obey Mrs. H. Gren. So if they ask you why viruses are non-living, you need to say they don't carry out homeostasis, they don't respire, they don't excrete, okay? Just make sure you pick out something from Mrs. H. Gren here to explain why viruses are non-living. In terms of their structure, they're very small, much smaller than bacterial cells. They're made up of a protein coat surrounding either DNA or RNA, and crucially, viruses are always pathogenic. They are always disease-causing. Examples include HIV, which infects our lymphocytes, reducing our body's ability to respond to pathogens. Tobacco mosaic virus, TMV, infects tobacco plants. It gives a characteristic mosaic pattern to the leaves. That yellowing indicates damage to the chloroplast and chlorophyll, meaning that the tobacco plant can't photosynthesize as well, meaning that it's less able to produce glucose, it can't grow as well. Viruses are only able to be produced inside another living organism. All living organisms share characteristics. State 2 characteristics that all living organisms share. So pick anything from Mrs. H. Gren. You could say that they all respire and excrete. That's worth 2 marks. Some organisms are pathogens. Which of these organisms cause a bacterial disease in humans? Read that super carefully. Okay, Chlorella, no, because it is a Protoctis. So already we don't have a bacteria here. Lactobacillus bulgaricus, yes, we have a bacteria, but that's used in yogurt making, so that's not going to cause a disease. So always work out why things are wrong. Mucor is a fungi, so we have issues there. It's not a bacteria. Pneumococcus, yes, it's a bacteria. It causes the disease pneumonia. So I'm happy that I've met the requirements for this question. Viruses are pathogens but not living. Describe the effect of a named virus that infects plants. Tobacco mosaic virus is what you want to mention here. You can write TMV if you prefer. And as I taught you in my video, it causes discoloration to leaves, meaning less photosynthesis occurs. Give 3 differences between the structure of viruses and bacteria. So make sure you clarify which one you're talking about. Viruses are smaller. Viruses have a protein coat. Bacteria have a cell wall. You can write the full comparison if you want. Viruses are smaller, bacteria are larger. Viruses have a protein coat. Bacteria don't have a protein coat. Bacteria have a cell wall. Viruses don't have a cell wall, but what I've written there is actually enough. Moving on to cells, tissues and organs. So we're going to start by listing a whole lot of definitions. Remember, you're going to use the same scaffold for all these definitions. So if we start with a cell, it's a group of similar organelles working together to perform a particular function. And it is that series of words we're going to use as our scaffold, but just changing a word each time as we move through the definitions. So if we take a tissue now, well, that is a group of similar cells working together to perform a particular function. An organ is a group of similar tissues working together to perform the same function. An organ system, well, it's a group of similar organs working together to perform the same function. So if we list some examples of organ system, could be the digestive system, which is made up of a group of similar organs, so stomach, esophagus, small intestine, large intestine, pancreas. Another type of organ system could be the reproductive system, that consists in a female of ovaries, fallopian tube, uterus, cervix, vagina. Moving on to differentiation. So differentiation is the term used to describe when cells become specialized. So for example, when they become nerve cells. A stem cell is a cell which has the ability to divide many times whilst remaining unspecialized, undifferentiated. There are 2 types of animal stem cell you need to be aware of. The first one is embryonic, the second one is the adult stem cell. As the name suggests, embryonic stem cells are found in embryos, particularly the early stage of development of embryos. They have the ability to differentiate or specialize into any cell types. So immediately you can see the added advantages of an embryonic stem cell. It can become any type of cell you want. For example, a bone cell, a hair cell, a nerve cell, a muscle cell. Moving on to adult stem cells, they're found in very specific places, namely the bone marrow, the skin and the lining of the small intestine. Although they have the ability, like all stem cells, to divide many times, unfortunately they can only become one specific type of cell. So for example, stem cells found in the bone marrow can only become blood cells. Stem cells have lots of uses in the treatment of disease, and this is known as stem cell therapy. A good example of stem cell therapy is in the treatment of leukemia, which is a type of blood cancer. So when the person is treated with both chemotherapy and radiotherapy, you find that both the cancerous cells and healthy cells are destroyed. So that's great, you've removed the cancer, but a person obviously really needs their white blood cells. So stem cell therapy involves introducing stem cells which are able to replicate and differentiate in order to replace those lost cells. Parkinson's disease is another good example of a disease that can be treated using stem cell therapy. In Parkinson's disease, nerve cells are destroyed, so it therefore makes sense that stem cells can be used to replace those damaged nerve cells. Ideally speaking, you'd use a person's own stem cells in stem cell therapy. The advantage here is that they're readily available. You have a complete tissue match, which means no chance of rejection by your own cells. And from an ethical point of view, it's great because there's no need to use embryonic stem cells. So if we consider the ethics now associated with embryonic stem cells, these have to come from aborted or miscarried fetuses. So obviously huge ethical concerns there. But the advantage of embryonic stem cells, as I've already pointed out, is their ability to specialize into any cell type. This puts them at a massive advantage compared with an adult stem cell, where I've already said there's only very specific places where you find adult stem cells. So this is why when people have horse riding accidents and they paralyze themselves from the neck down, you tend to find they remain paralyzed for life, and that's because you don't have stem cells in your spine. However, embryonic stem cell therapy is a way of potentially making more nerve cells in order to repair those damaged nerves in your spine. And I'm just going to drop a table from my revision guide into this video so you can look at the various advantages and disadvantages associated with the use of stem cells. As an embryo develops, its cells differentiate. Explain the importance of cell differentiation in the development of the growing embryo. So clearly the embryo needs a whole bunch of different types of tissue, ones which will become heart tissue, muscle tissue, bone tissue, etc. So we need cell differentiation because that allows the cells to become specialized in order to produce those tissues. Which of these is a feature of adult stem cells? They do not divide, that's not true. They divide by meiosis, no, they divide by mitosis. They can become all cell types, no, that's only a feature of the embryonic stem cell. They're found in some tissues and organs, yes, that's correct, such as the bone marrow. Adult stem cells or embryonic stem cells can be used in medical treatments. Explain why the choice between these two types of stem cells can cause issue for doctors. Let's differentiate the ethical issues here. So the embryonic stem cells have ethical concerns because after all we know that those embryos are potential human life, but the advantage of them is that they can differentiate into any cell type. Moving on to biological molecules. So what elements are lipids made up of? Remember, lipids are fats. So lipids contain carbon, hydrogen and oxygen. A lipid molecule is arranged in the following way. It's made up of 3 fatty acids and a glycerol. To test for the presence of lipids, you want to add ethanol and water to your sample, give it a good shake, and if lipid is present, you'll see a milky white emulsion. Moving on to proteins now, you'll get the carbon, hydrogen, oxygen elements, but in addition, you'll also find that there's nitrogen and sometimes even sulfur. Proteins are made up of long chains of amino acids. In order to test for the presence of protein, you want to add bioarray reagent. If there's no protein, you'll see a blue color. If protein is present, you'll see a purple color. Some good examples of proteins found within the human body include collagen and keratin found in the skin, all enzymes, and hemoglobin found in red blood cells. That hemoglobin, remember, binds to oxygen. Moving on to carbohydrates. Carbohydrates contain only carbon, hydrogen, and oxygen. A good example of a carbohydrate is glucose. Remember, to test for glucose, you add Benedict's reagent to your sample and you need to heat it in a water bath. You'll see a range of potential colours. Blue indicates no glucose is present, whereas brick red indicates that lots of glucose is present, but you could also get a range of colours. So any colour that appears nearer the blue end indicates low glucose presence. Anything that appears near the redder end, so orange, yellow, indicates a higher glucose presence. Storage compounds of carbohydrates include glycogen and starch. Remember, these are long chains made up of simple sugars like glucose. In order to test for insoluble starch, this time we're going to add iodine. If no starch is present, you'll see a brown colour. If starch is present, you'll see a blue-black colour. A student investigates the sugar content of 4 different fruit juices. This is the student's method. Add 5 centimeters cubed of fruit juice to a boiling tube. Add 5 centimeters cubed of Benedict's solution to the boiling tube. Place the boiling tube in a water bath at 70 degrees Celsius for 3 minutes. Remove the boiling tube and record the colour of the solution. The student uses this method for each of the 4 fruit juices. The table shows the student's results. So let's have a look. We know that Benedict's tests for glucose. If we look down the results, you can see that fruit juice A contains no glucose because the Benedict solutions remained blue, whereas fruit juice B contains lots of glucose because it's turned brick red. Yellow and green, these still contain glucose, but not as much as B. So like medium levels of glucose for C and low glucose for D. Give 2 variables that the student controls in his investigation. So what is he keeping the same? the volume of the fruit juice, and the volume of the Benedicts. Give the juices A, B, C and D in order of sugar content from highest to lowest. Happily I've already done that, so I said that B has the most, followed by C, then D, and then lastly A, which has none at all. The student is now given sugar solutions with concentrations of 1%, 5%, 10% and 20%. Explain how the student could use these solutions to estimate the concentration of sugar in the 4 fruit juices. So basically we're going to repeat the same experimental method as before, this time with the various concentrations of sugar solution. We'll look at the colour change that occurs, and then obviously if the colour matches fruit juices A, B, C or D, we'll be able to estimate how much sugar they each contain. So let's use our methodology from before. Now we're moving on to enzymes. First of all, defining the term metabolism. This is the rate at which chemical reactions take place in our body. The role of enzymes within metabolic reactions is that they act as catalysts. They are biological catalysts which speed up the rate of a reaction without being used up, and they're chemically unchanged. What distinguishes enzymes from other catalysts is this use of the word biological. They're biological because enzymes are made out of protein. Focusing in on some key enzymatic terms. So what is the substrate? That is the substance upon which the enzyme acts. The active site is the special site on the enzyme where the substrate binds. When that substrate binds, you have what's known as an enzyme substrate complex, and then once the enzyme's finished its work, the substrate is released as a product. Optimum is a word used to describe when the enzyme is working at its highest rate. And then another key term is the word denature. Remember, if the temperature is too high or the pH alters too significantly, the enzyme denatures, which means that its active site changes shape and the substrate no longer binds. Remember that enzymes are highly specific, so each enzyme will only fit one type of substrate. It's highly complementary. The substrate shape is complementary to that of the enzyme's active site. If we now consider the effect of temperature on enzyme activity, as you increase the temperature initially, the enzymes and substrate molecules have more kinetic energy. This means that collisions between the two occur more frequently, so you end up with more enzyme-substrate complexes being formed. The enzyme's working at its highest rate here, and we call this the optimum temperature. If the temperature increases above that, what happens is the enzyme's active site is altered. The enzyme is denatured, meaning that the substrate no longer fits. Additionally, enzymes have optimal pHs, such as the enzyme protease found in the stomach. Its optimum pH is going to be a very low pH because it needs a highly acidic environment around pH 2. So if you increase that pH further, the enzyme denatures and its active site changes shape. Moving on to the movement of substances into and out of the cell. So there are 3 different types of transport we need to consider. Number 1, diffusion, 2, active transport, and 3, osmosis. So diffusion is the simplest. It is simply the net movement of a substance from an area of high concentration to an area of low concentration. This is a passive process and does not require the input of energy or ATP. You could be asked the factors which increase the rate of diffusion or affect the rate of diffusion. So the steepness of the concentration gradient. The steeper the concentration gradient, the faster the rate of diffusion. Surface area also plays a part. The larger the surface area, the faster the rate of diffusion. A high temperature will increase diffusion because the molecules have greater kinetic energy. A shorter diffusion distance means that molecules have less far to travel, again, so you'll have a higher rate of diffusion. Unlike diffusion, active transport requires an input of energy. So if we look at its definition, it's the net movement of molecules from an area of low concentration this time to an area of high concentration against a concentration gradient, therefore requiring ATP or energy. Active transport is vital in living organisms because it enables the movement of molecules against the concentration gradient A good example of this is the uptake of glucose in the small intestine. This occurs by active transport. Moving on to water now. So the movement of water molecules is known as osmosis. So our definition of osmosis is as follows: the net movement of water molecules from an area of high water potential to an area of low water potential across a partially permeable membrane. So osmosis is similar to diffusion in that we're going down our concentration gradient, but crucially it involves the movement of water molecules and requires a partially permeable membrane. A solution which has a high water potential just means that it contains a large number of water molecules. It is in essence dilute. A low water potential solution is one with few water molecules, so could be quite high in solutes, and therefore is what we would describe as a concentrated solution. Remember, it's important that we can move water around in living organisms because water is a valuable solvent. It supports many chemical reactions that take place in our cells, and it enables dissolved substances to be moved around an organism. Osmosis involves the use of a partially permeable membrane, and the key word here is partially. It only allows certain molecules to pass through. Larger molecules are trapped and are not allowed to move. If we consider osmosis in practice, what happens if we place an animal cell in a high water potential solution, for example, pure water? Well, what happens is based enormously on the fact that an animal cell has no cell wall. So if you place an animal cell in pure water, water enters by osmosis, according to our definition, from an area of high water potential in the surrounding solution to an area of low water potential inside the animal cell. Across that partially permeable membrane, the water enters the animal cell and it keeps swelling and expanding until it can take no more and it bursts. And then we call this, if it occurs to red blood cells, hemolysis. If you were to place your red blood cell in a highly concentrated solution, so one with low water potential, this time water would leave your animal cell by osmosis, causing it to become shriveled and flaccid in appearance. Let's understand what happens when a plant cell is placed in pure water. So always ask yourself where there's more water, where there's a higher water potential. Pure water by definition has the highest water potential. So if you think about which way that water moves, it's going to enter the plant cell. So water enters the plant cell by osmosis from an area of high water potential. So do define osmosis here and specify where that higher water potential is. So that will be the surrounding solution. To an area of lower water potential, which will be inside the plant cell. Across a semi-permeable membrane. And then the plant cell itself becomes turgid, which means swollen. What about if the plant cell was placed in salt solution? This time that salt solution will have a lower water potential. Water always moves from an area of high water potential to an area of low water potential. So in this case, we would change the wording so that water leaves the plant cell because it's moving by osmosis from an area of high water potential, this time inside the cell, to an area of low water potential, which will this time be the surrounding solution. And because water's left that plant cell, it will become flaccid and plasmolysed, which is when the cell membrane pulls away from the cell wall. So make sure you can discuss those two different scenarios. So if we have a look here at the diagrams from my revision guide, here's our plant cell. We've placed it in pure water, so water's entered the plant cell. The plant cell has swollen up, it's become turgid. In this situation, the plant cell's been placed in a salt solution with a lower water potential, meaning that water leaves the plant cell, so it starts to shrivel up and plasmolyze, so that cell membrane has pulled away from the cell wall. Here you can see a comparative scenario with a red blood cell, which is an example of an animal cell. Whereas with the plant cell, it became turgid and it didn't burst because of the presence of that supportive and protective cell wall. In the case of an animal cell, when it's placed in pure water, the water keeps flooding in by osmosis. There's no cell wall, so eventually the cell will burst. They could ask you about an investigation looking at the effects of different concentrations of salt solution on plant tissue. So the different concentrations, because that's what we're changing, that will be our independent variable. And if you're asked to specify, pure water would be 0.0 mole, and this is gradually becoming saltier. We're going to use cylinders of potato chips, which do need to have an equal size, so that's a good control variable. In terms of what we're measuring, we'll look at the change in mass of those potato chips, so that's our dependent variable. Do make sure that you dry the potato to ensure that we don't have any excessive water attached. We place our cylinders into a test tube of known salt solution. It's up to you a time frame. You might want to go longer than that, maybe even up to 12 hours. And then what you want to do next is find the percentage change in mass. They could ask you to calculate that. It's change in mass over original mass times by 100. And the reason why you want to calculate the percentage change in mass is because it's hard to actually guarantee that the initial masses are all the same. So by calculating the percentage change in mass, it enables you to make a valid comparison. You always get a mark for writing that. And then they'll often ask you to plot a graph. Here we can see on our y-axis our percentage change in mass, so our dependent variable, what we're measuring. Our x-axis is what we've changed, our independent variable, so the concentration of salt solution. Down here you've got pure water. Up here you've got a much saltier solution. If you plot your results, this point here is incredibly important because here the percentage change in mass is 0, meaning that there's no net increase or decrease in the mass of the potato. So that automatically tells us that the water potential in the potato must be the same as the surrounding solution, which for this potato chip is 0.3 molar. So do make sure you can pick that point off a graph. This apparatus can be used to show osmosis, explain what happens to the level of the sucrose solution in the glass tube. So here's our partially permeable membrane. Here's a surrounding solution containing water, which means it has a high water potential. Sucrose solution contains a lot of sugar, which will mean that this solution has a lower water potential. So if you think about osmosis occurring, there's the partially permeable membrane we require for osmosis. In this particular situation, osmosis is going to occur in this direction. Water will move from the surrounding solution into the glass tube, meaning that that level here will increase. So we need to say why that level increases, because water enters the tube by osmosis from an area of high water potential. It's very similar to the explanations I was just providing. Say where that is in the beaker to an area of lower water potential, which will be inside the glass tube. Describe how this apparatus could be modified to measure the rate of osmosis at different temperatures. So how should we change that temperature? Use a water bath at 5 degrees, 10 degrees, 15 degrees, etc. And because we're after the rate of osmosis, which always involves a time implication, we want to look at the level in that glass tube over a particular time period. So measure the height of solution in the glass tube and do provide suitable apparatus using a ruler after 1 hour, timed with a stop clock. Plant nutrition. So clearly plants don't feed in the way that animals do. So how do they obtain their food? Well, they obtain it through the number one plant process, which is photosynthesis. Plants are incredibly clever in that they're able to take carbon dioxide, react it with water, and in the presence of light, they can produce their food, glucose, and they produce oxygen as a byproduct. And that is your word equation for photosynthesis. Make sure you can also state the symbol equation. It's useful to remember that you need a 6 in front of all the formulae apart from glucose. So that means 6 CO2 reacts with 6 H2O to form glucose, C6H12O6 plus 6 O2. Now, where does photosynthesis take place? It takes place in the chloroplast of the plant cell. But clearly, not all plant cells contain chloroplasts. After all, ones found within the root aren't exposed to light, so you won't find chloroplasts there. Most of the chloroplasts are actually found in the palisade mesophyll part of the leaf, and we'll talk a lot about the leaf structure and its adaptations for photosynthesis a little later. One thing they could ask you about is the limiting factors which affect the rate of photosynthesis. So this is a factor in a reaction which is in the shortest supply, and a lack of this factor means that the rate of photosynthesis no longer increases. There's 3 limiting factors they want you to be able to state. That's temperature, light intensity, and carbon dioxide concentration. If we take different parts of the day, we'll be able to understand which factor is most likely to be limiting. So early in the morning, clearly it's not the hottest part of the day, so low temperature is likely to be the limiting factor here, and it means that the enzymes involved in photosynthesis have less kinetic energy and are less able to catalyze the reactions involved. At midday, temperature is no longer the limiting factor. After all, it's the hottest part of the day. Here, carbon dioxide is likely to be the limiting factor. Let's look at the leaf structure in a great amount of detail now. So in terms of its adaptations for photosynthesis, first of all, start by stating that it is flat and has a large surface area. This means it can absorb as much light as possible. If you also think about leaves, they're very thin, and that means that there's a short diffusion distance for the gases needed in photosynthesis to diffuse. Starting with the topmost layer, the waxy cuticle is present in order to prevent water loss by transpiration. Next up, we have the upper epidermis, which is transparent to allow light to enter the leaf. The palisade mesophyll contains the layer of cells which most closely resemble what you'll be familiar with in terms of a plant cell. Here you'll find lots of chloroplasts which absorb sunlight in photosynthesis. This is where the glucose is made. Then you have the spongy mesophyll, which is characterized by air spaces which allow gases needed for photosynthesis to diffuse. So really we're talking about carbon dioxide here. But how does carbon dioxide enter the leaf? Well, it enters through small holes located on the lower surface of the leaf, known as stomata. When those holes open, carbon dioxide is able to diffuse into the leaf. At the same time, water leaves, and this process also occurs by diffusion. It's not osmosis because there's no partially permeable membrane in the stomata. One thing to be aware of is that if the water loss, the transpiration, is occurring too quickly, the plant may start to wilt. So at this point, special cells known as guard cells will cause the closure of the stomata, meaning that no more water leaves, but at the same time it means no more carbon dioxide can enter, therefore limiting how much photosynthesis can occur. One investigation which is frequently asked about is how do we investigate the effect of light intensity on the rate of photosynthesis. So our independent variable, what we're changing, will obviously be our light intensity, and the most straightforward way of altering this is by simply holding a lamp at different distances away from our plant. For example, 10 centimeters, 20 centimeters, 30 centimeters, etc. But how are we going to measure the rate of photosynthesis? Now, when you're asked about the rate, there's always a time implication. So we need to look at how much photosynthesis is occurring in a particular time frame. The easiest way to measure the rate of photosynthesis is by looking at how quickly one of the products of photosynthesis is produced. And if you remember, photosynthesis produces glucose and oxygen. And the easiest thing to monitor is the rate of oxygen bubble production. And we can simply do that by either counting the number of oxygen bubbles released from pondweed in 60 seconds, for example, or we can measure that volume more accurately by using a gas syringe. We like to use pondweed because it exists in water, so it makes those oxygen bubbles far more visible. So our dependent variable here could be the number of oxygen bubbles released in 60 seconds. What about our control variables? Well, clearly we need the same length of pondweed, the same species of pondweed, the same carbon dioxide concentration, and the same temperature. The faster the bubbles are released, the greater the rate of photosynthesis, and we'd obviously expect more photosynthesis to occur when the lamp is closer to the pondweed. Moving on to testing a leaf for starch. Now, plants don't store glucose, instead they convert it into insoluble starch. The reason for that is because starch is insoluble, so it has no osmotic effect. Now, in order to force a plant to use up its starch, you need to stop it photosynthesizing, and the way to do that is by placing it in the dark for a minimum of 12 hours. So how do we test a leaf for starch? Well, the first thing you need to do is boil the leaf in order to kill it. We need to remove the green from the leaf, and the green is due to the chlorophyll. So how do we remove the chlorophyll? We need to boil in ethanol. Do be mindful that ethanol is highly flammable. We then wash the leaf in cold water, and then finally, to test for starch, remember from a previous part of the specification, we use iodine here. Iodine stays brown when no starch is present. It turns blue-black when starch is present. 2 mineral ions to be aware of. Now, mineral ions are absorbed at the roots, specifically the root hair cells. The role of the nitrate ions that the plants absorb is to help build plant protein. The role of magnesium ions is to produce chlorophyll. A student does an experiment to show that a leaf is only able to produce starch if it receives enough light. The student removes all the starch from the leaf before starting the experiment. Describe how the student could remove all the starch from the leaf. So you need to make that leaf use up its starch, and the only way it will do that is if it can't photosynthesize and make glucose, because it's going to use that starch to produce the glucose it needs. So how do we stop it photosynthesizing? Place leaf in the dark. And the second mark here is for providing a long enough time frame for 12 hours. State why the student removes all the starch from the leaf before starting the experiment. That's to prove that any starch present was made during the experiment. Before testing a leaf for starch, chlorophyll needs to be removed. Give a safety precaution the student needs to take when removing chlorophyll. So they're expecting you to know an awful lot about this experiment. Obviously, we need to boil the leaf in order to kill the leaf, but then remember that we boil in ethanol in order to remove that chlorophyll. So what's the safety precaution? Well, ethanol is flammable, so it will make sense to use a water bath rather than a direct flame. Give a suitable control the student could use in this experiment. So we know that in the experiment the leaf is going to be exposed to light, so the control would be to keep the leaf in the dark. Describe how the structure of the leaf is adapted for photosynthesis. So let's go through all the various adaptations. So we're going to start by saying the leaf is flat and has a large surface area to absorb as much light as possible. Let's go through the various layers. So the upper epidermis is transparent to allow light to enter the leaf. The next layer is the palisade mesophyll, which, remember, contains lots of chloroplasts which carry out photosynthesis. Moving down, we have the spongy mesophyll, which contains air spaces that allow that all-important carbon dioxide needed for photosynthesis to diffuse through the leaf. Moving down again, those holes are called stomata. Allows CO2 to enter the leaf. And what's the other reactant of photosynthesis? So remember, we have 6 CO2 reacting with 6 H2O to form glucose and oxygen. So we've talked about how that carbon dioxide is entering. What about the water? Xylem in the vein of the leaf allows the entry of water to the leaf. Let's define the term balanced diet. A balanced diet provides all the nutrients in the correct amounts needed to carry out life processes. And this is important in order to maintain our good health. So what are the components of a balanced diet? We're going to go through the various nutrients, which foods contain those nutrients, why the nutrient is required by our bodies, and any appropriate deficiency diseases. So starting with carbohydrates, these are found in things like bread and pasta, and the reason why we need to consume carbohydrates is because they're an important energy source. Moving on to fats, found in butter, cream, etc. Fats are a more concentrated energy source and they actually help insulate our body, and it explains why mammals like seals and whales have an awful lot of fat, because they live in cold water and it helps them to stay warm in those cold waters. Protein is found in meat, in lentils. It's needed for the growth and repair of our muscles, and if we don't eat enough protein, we can suffer from horrible deficiency diseases such as kwashiorkor or marasmus. Everyone knows we need to drink water, but why do we need it? It supports the chemical reactions that take place in our cells. It acts as a solvent. Fibre now, found in fruits and vegetables. Fibre provides roughage. And what that means is it helps food move through our digestive system in a process known as peristalsis. So a good thing to write in your exam is that fibre aids peristalsis, and by doing so it prevents constipation. Let's move on to various minerals, including calcium, found in dairy products like milk. Calcium is needed for strong teeth and bones, and if you don't get enough calcium in your diet, you're likely to suffer from weak teeth and bones. Iron is found in red meats, spinach. Iron is an important component of the hemoglobin in red blood cells, and as a result, we need iron to help us transport oxygen around our bodies. Without enough iron, you can suffer from a deficiency disease known as anemia, where symptoms include tiredness and fatigue. Various vitamins now vitamin A, found in fish liver oils. This is needed to help with our vision, and people who have insufficient quantities of vitamin A suffer from night blindness. Vitamin D, also found in fish liver oils, it's also made by the action of sunlight on our skin, is also needed for strong teeth and bones. Without enough vitamin D, you can suffer from rickets, which is when your legs start bowing out. And then lastly, vitamin C, found in citrus fruits like oranges and lemons. This actually helps to stick together the cells which line your mouth, and if you don't eat enough vitamin C, you might suffer from an affliction known as scurvy, where your gums start bleeding, and historically sailors are famous for suffering from scurvy, so in the end, the boats they used to travel on used to contain barrels of oranges and lemons. Make sure you're aware of the differences in the type of diets people require based on their activity, their lifestyle, etc. So people with more sedentary jobs will obviously require less food, so less carbohydrates, less protein, less fat, compared with people with very physical occupations. You often find that when girls reach puberty that they suffer from anemia, and that's due to a lack of iron brought on through menstruation, which remember is the blood loss associated with a period. Pregnant women will obviously need more of everything because they need to support the growing fetus in terms of its muscle growth, bone development, production of blood, so obviously we'll need more iron, more calcium, more protein, etc. in order to support that growing fetus. Cow's milk contains a mixture of nutrients including protein, fat, sugar, vitamins and minerals. The main sugar in milk is a carbohydrate called lactose, which chemical elements are present in carbohydrates. So that's CHO, carbon, hydrogen and oxygen. Let's look down at the options. It's B. Describe how to test a sample of milk for protein. Keep this really minimal. Use bioarray reagent. The sample turns purple in the presence of protein. Some people drink a milk alternative produced from plants such as soy and rice instead of milk produced by animals. The table shows nutritional information for different milk products. So we have cow's milk, soy milk, rice milk, together with the energy, how much protein they have, how much fat, carbohydrate and calcium. The recommended daily amount of calcium for a child is 1400 milligrams. Calculate the mass of cow's milk that would contain 1400 milligrams of calcium. So make sure you're looking along the cow row. So there's 250 milligrams in 224 grams of milk. So take that 1400, divide it by 250 and then multiply it by 224. 1254 grams. Discuss whether soy milk or rice milk is a suitable replacement for cow's milk in the diet of children. So we need to look at both the soy and the rice and decide based on the nutrient contents if it's a good replacement for cow's milk. Remember, we also need to take each of these nutrients and understand why they're important. So if we start with the energy, we can see that both soy and rice have less energy in them compared with cow's milk. In terms of the protein, we can see that the soy has the same mass of protein, which means that soy milk will be suitable for helping to grow and repair muscles. Rice has very little protein, so that's going to mean that we don't have enough protein, meaning you can end up with certain diseases like kwashiorkor. What about the fats? Well, it depends which way you look at it. There's less fat in soy and rice, which is good in terms of there won't be as much weight gain, which could lead to obesity. However, fat's also important, remember, as an energy source and for insulation. Carbohydrates, soy has less carbohydrate, rice has more carbohydrate. So here rice is a good energy source. Now if we look at calcium, soy has a huge amount of calcium actually, so that's going to be really good for our teeth and bones. Rice is going to be a very poor substitute, seeing as it has no calcium in it whatsoever. So overall, I would definitely pick soy as opposed to rice as my replacement milk product if I weren't drinking cow's milk anymore. Defining digestion now. Digestion is the breakdown of large insoluble food molecules into small soluble ones, which can then be absorbed through the wall of the small intestine. There are two types of digestion. The first one is mechanical, the second one is chemical. So mechanical really means just breaking down that food into smaller pieces. There's no change in the chemical structure of the food. So if you think about it, the first place that mechanical digestion takes place is in your mouth. Your teeth chew, breaking up that food, increasing its surface area. A second place mechanical digestion takes place is in your stomach, where the stomach muscles contracting help to break down that food further, and hydrochloric acid in the stomach also assists with this. Chemical digestion really means the altering in the structure of the food. It means making that food more soluble, and it's enzymes which are responsible for this. So in the mouth, we've already discussed the mechanical digestion by the teeth, but also the enzyme amylase. This will be responsible for chemical digestion, the digesting of starch into maltose. Food then passes down the esophagus, so the bolus, the ball of food, passes down the esophagus with the aid of peristalsis, which is the contraction of muscles. Food reaches the stomach. I've already mentioned what the stomach does. Remember that hydrochloric acid also lowers the pH, meaning that any bacteria ingested will also be destroyed. Protease is an enzyme secreted by the stomach. It helps break down proteins into amino acids. Further peristalsis occurs in the small intestine. Remember that fiber is essential in your diet because it provides bulk to your food, it provides roughage, it aids the process of peristalsis, therefore helping prevent constipation. Moving on to bile. So remember, bile is produced by the liver, stored in the gallbladder and released into the small intestine. The role of bile is twofold. Number one, it emulsifies fats, increasing their surface area, meaning that the enzyme lipase can work more effectively. Number two, it neutralizes the pH of food, meaning that enzymes released into the small intestine aren't denatured. We've already mentioned the enzymes amylase and protease. Remember the enzyme maltase, this breaks down maltose into glucose. You've got the enzyme lipase, this breaks down lipids into fatty acids and glycerol. The pancreas secretes all three enzymes into the small intestine. Now the small intestine's role is to absorb those soluble food molecules. It's adapted in order to increase the rate at which absorption takes place. The microvilli and villi increase the surface area, meaning that absorption occurs more quickly. You've got the presence of a lacteal that aids fat absorption. You've got a good blood supply, lots of blood capillaries. Thin walls provide a short diffusion distance. The role of the large intestine is to reabsorb water. By this point, you'll have reabsorbed all your soluble food molecules, leaving you with just the indigestible foodstuff. This makes up the bulk of feces, which are stored in the rectum and released into the anus. Let's discuss some key definitions now. So ingestion, effectively eating, that's the intake of food. You've got egestion, which you mustn't confuse with excretion. Now, egestion is the removal of feces from the anus, so in effect pooping. Excretion is the removal of metabolic waste. Assimilation is the buildup of large molecules from small molecules. Digestion I've already mentioned. Absorption I've mentioned in passing. It's the movement of soluble food molecules across the lining of the small intestine into the blood. Now we're going to describe an experiment to investigate the energy content of food. So taking, for example, a piece of popcorn. The first thing you need to do is measure the mass of the popcorn using a balance. Add 50 centimeters cubed of water to a boiling tube and measure its initial temperature using a thermometer. You want to set up your clamps down to allow you to hold that boiling tube. Then you're going to set the popcorn alight. Make sure that popcorn's been placed on a mounted needle, as that will make it easier to hold. So the popcorn starts burning. Immediately you want to hold it underneath the boiling tube because the heat coming off that popcorn will raise the temperature of the water. Do make sure that that popcorn burns entirely. You want complete combustion to take place. That means burning in a plentiful supply of oxygen. And then at the end of the experiment, you want to take the final temperature of that water. You'll then be able to calculate a temperature difference, and then to calculate the energy content, remember we use the equation Q equals mc delta T, where m is the mass of water being heated, C is the specific heat capacity of water, which is 4.2, and delta T is your change in temperature that you've just calculated. Right, let's go through a past paper question. So obesity is caused when energy input is greater than energy output. A student likes to eat potato crisps but is concerned about obesity. The student has a choice of 2 different types of crisp to eat. Describe an experiment the student could use to determine which type of crisp contains the least energy. So obviously the equation we need to use is E equals mc delta T, because E after all is the energy. And we can calculate the energy by finding the mass of the crisps. Do make sure, because you're using 2 types of crisp, make sure you're using the same mass for each crisp. C is the specific heat capacity, which has a certain value. It's 4.2 for water. And then we also need to find the change in temperature. But what are we finding the change in temperature of? Well, remember, we're going to use that burning crisp to heat water in a boiling tube. So we're going to be measuring the change in temperature of that water. Clearly, we need to set the crisp alight. We're then going to use the burning crisp to heat the water. I've specified a particular volume of water that is worth a mark. And we need to keep heating that water until we've burnt that crisp completely. And then in terms of reliability, do make sure you repeat and calculate an average. Respiration now. So aerobic respiration involves the use of oxygen. The word equation is oxygen plus glucose goes to carbon dioxide plus water plus the release of energy. Remember that energy is always released, it's never produced. Make sure you can write the symbol equation as well. So 6s are important here with the balancing. You want 6O2 plus C6H12O6, which is the formula of glucose, goes to 6CO2 plus 6H2O. Although energy is released, ATP is made, and it's this ATP which is used to transfer the energy. What is this energy released by respiration used to do? Well, it's used to help us contract muscles, move substances by active transport, carry out assimilation, which is building up large molecules from small molecules. It's also used in cell division as well as the manufacture of more organelles found within our cells. We've already said that we need oxygen for aerobic respiration. How does oxygen enter our body? Well, it enters through an inhalation, that's a breath in. The oxygen actually enters our blood in our lungs. Remember that General atmospheric air contains around 20% oxygen. It's mostly made up of nitrogen with some argon gas, carbon dioxide and water vapour. If we compare air that we breathe in, so that's inhaled air, compared with air that we breathe out, exhaled air, it makes sense that we absorb oxygen into our bodies, so therefore exhaled air will contain less oxygen. Because carbon dioxide is produced by respiration, exhaled air will contain more carbon dioxide and more water vapour. So make sure that it makes sense to you. If you look at the equation for aerobic respiration, exhaled air must therefore contain more CO2, more water and less oxygen. Hydrogen carbonate indicator can be used to demonstrate how much carbon dioxide is present. So atmospheric air contains around 0.04% carbon dioxide. This turns hydrogen carbonate indicator orange. So if you see that that indicator is orange, you can say that there's around 0.04% CO2. High levels of CO2 turn the indicator yellow. Low levels of CO2 turn the indicator a red-purple colour. Anaerobic respiration can occur if there's insufficient or a lack of oxygen. Here the glucose is incompletely broken down and it actually releases less energy and produces less ATP compared with aerobic respiration. Anaerobic respiration is carried out by yeast cells as well as our muscle cells if we experience intense periods of exercise. When yeast anaerobically respires, it takes that glucose and converts it into ethanol and carbon dioxide. This process is actually used in the bread making industry to help bread dough rise. The ethanol produced is used in the alcoholic drinks industry, so to make beer and wines. When our muscles anaerobically respire, this time the glucose is converted to lactate. Anaerobic respiration can cause muscle fatigue over time, and that's due to what's known as an oxygen debt. It means that when you stop exercising, your breathing rate stays high, and the reason for that is because you need to introduce more oxygen into our body in order to break down the lactate produced by anaerobic respiration. A student investigates the effect of temperature on the rate of anaerobic respiration in yeast. The student measures the rate of gas produced in centimeters cubed per minute. The graph shows their results. Name the gas produced by yeast during anaerobic respiration. Remember, that is carbon dioxide. We use it in our alcohol making industry. Explain the effect that increasing temperature has on the rate of gas production by the yeast. So let's talk about the various portions of the graph. I'm going to call them A, B and C. And because they're asking us for an explanation, we need to give the biological reasons as to why you see an increase here in terms of gas production. This is the peak, and then you see it decrease down here. So I'm going to say at A, remember these answers are always to do with enzymes. Enzymes have more kinetic energy, meaning more enzyme substrate complexes form. In biology, if they're asking about temperature, it's always to do with enzymes. At B, let's double check the optimum temperature has been reached, which from the graph is approximately 37 degrees. And then at C, the temperature is too high. And remember what happens to enzymes when they're too hot? They denature, which means their active sites change shape and the substrate no longer binds. Describe how the student can measure the rate of gas production in this experiment. As I keep saying, rate has a time implication, so we need to look at the volume of gas produced in a particular time frame. Measure the volume of CO2 in 60 seconds. And how are we measuring that volume? Using a gas syringe. Gas exchange in plants. As the name suggests, this is all about how both oxygen and carbon dioxide move into and out of the plant. This is going to be happening specifically at the leaf, and remember we looked at all the various layers of the leaf. The small opening in the leaf is known as a stomata. Remember, it's just a pore. It can open and close based on whether the guard cells surrounding it are swollen or flaccid, and both carbon dioxide and oxygen can move into and out of the leaf via the stomata. But what direction do these gases move in? Well, it totally depends on what process is occurring. So respiration, which occurs all the time, Remember the equation for respiration: oxygen plus glucose produces carbon dioxide and water. So when the leaf is respiring, oxygen will diffuse from the atmosphere into the leaf via the stomata, and carbon dioxide will be released as a byproduct. Photosynthesis, which obviously only occurs when it's light, requires carbon dioxide. So in this particular situation, CO2 will be diffusing into the leaf, and oxygen, a byproduct of photosynthesis, will diffuse out of the leaf. When we have dim light levels, the rate of respiration and the rate of photosynthesis are approximately equal, so you'll see no net movement really between the oxygen and carbon dioxide between the leaf and the surrounding air. How is the leaf adapted for gas exchange? Well, it has a large surface area. It's thin and flat, which provides a short diffusion distance for those gases to diffuse. We can investigate the rate of photosynthesis and the rate of respiration by using hydrogen carbonate indicator, which changes colour based on the amount of carbon dioxide present. Just to be aware, the atmospheric carbon dioxide sits at 0.04%, and hydrogen carbonate indicator will turn orange in atmospheric levels of carbon dioxide. It makes sense, therefore, that if the rate of photosynthesis equals the rate of respiration, as I've said, that there's no net movement of carbon dioxide, that means that the hydrogen carbonate indicator will turn orange, and we'd expect to see that in lower light levels. But what about when it's bright light, when the plant's photosynthesizing a huge amount? It's obviously going to be using up far more carbon dioxide than it's releasing in respiration, and as a result, the CO2 levels drop, the hydrogen carbonate indicator turns purple. At night, the plant can't photosynthesize, it's just respiring, so it'll be releasing much more CO2 than it's using. Higher CO2 levels are indicated by the hydrogen carbonate indicator turning yellow. And it's important that you can specify these various colours, what concentrations of carbon dioxide they represent, and what you can infer about the rates of respiration and photosynthesis as a result. Gas exchange in a flowering plant changes depending on conditions. Complete the passage by writing a suitable word in each blank space. Plants carry out photosynthesis to produce glucose. To enable this process to occur, the leaf cells absorb carbon dioxide and release oxygen. At the same time, the cells in the leaves are respiring. This means that they are using oxygen. You need to know those equations, and producing carbon dioxide. If the leaves are in bright sunlight, then the rate of photosynthesis will be greater than the rate of respiration. If the leaves are in dim light, then the rate of respiration will be greater than the rate of photosynthesis, and there'll be a net production of, obviously that dim light, we're not told how much light there is, so in this situation they're telling us the rate of respiration is greater, so use the wording in the question. So you're going to see a net production of carbon dioxide. In conditions where there's no net absorption or release of CO2, the rate of photosynthesis and respiration are equal, and the plant is at its compensation point. Describe how you could use hydrogen carbonate indicator to investigate the effect of light intensity on the net gas exchange in a leaf. So our independent variable, it's never a bad idea to use variables here. So what are we changing? Light intensity. How are we going to do that? They want a method. Is changed by holding a lamp ten centimeters away, 20, etc. Okay, what's our dependent variable going to be? So what are we measuring? We're going to be looking at the colour that hydrogen carbonate indicator changes. And you could give an example here. E.g. turns yellow when lots of CO2 is released. And we know that that's when more respiration is occurring compared with photosynthesis. Our control variables, what we keep the same. Use same species of plant and same temperature. So yeah, use your variables here and it will give you a scaffold to answer these type of questions. The respiratory system now, we'll start with its anatomy. So your windpipe, its scientific name is your trachea, which runs down here. Then it branches, quite similar to the branches of a tree really, into two bronchi. Smaller branches follow on, called bronchioles, and then it ends in air sacs, known as alveoli. You have a thick layer of muscle that sits towards the bottom of your thorax. That's called your diaphragm, which is able to contract and relax. Your rib cage provides a bony cage over the top of your lungs to help protect them. You've also got intercostal muscles, which are muscles which sit between your ribs, and they'll play an important role when you take a breath in and when you take a breath out. So I've already mentioned that the alveoli are air spaces. This is effectively where oxygen goes from your air in your lungs into your blood and carbon dioxide leaves your blood. There are small vessels known as capillaries which kind of cover your alveoli. They provide a very good blood supply in order to allow that gas exchange to take place. So what are the adaptations of the alveoli for gas exchange? Well, I've already said there's a large surface area, and that's due to their grape-like structure. They've got thin walls, so that provides a short diffusion distance. They're moist, which allows gases to dissolve, and they have good ventilation, which maintains a steep concentration gradient, and that means that diffusion of gases occurs much more quickly. Focusing in on the trachea. Now, the trachea is supported by rings of cartilage. They help to support the trachea, stop it from collapsing, and they also enable your neck to bend. So focusing in now on ventilation, that's the movement of air into and out of the lungs. What about an inhalation, a breath in? The following changes occur. First of all, your diaphragm contracts, it flattens, your external intercostal muscles contract, and together these cause an increase in the volume of your thorax. Because the volume's increased, by definition, the pressure must decrease, and that's really in comparison to atmospheric air, and as a result, air is drawn into the lungs. What about an exhalation now, a breath out? The opposite occurs. This time your diaphragm relaxes, it becomes dome-shaped. Your external intercostal muscles relax. Together, these reduce the volume within your thorax, meaning that the pressure this time increases compared with atmospheric pressure. Therefore, air is forced out of your lungs. If you consider the difference in composition between inhaled and exhaled air, clearly inhaled air will contain more oxygen compared with exhaled air because the oxygen's been used by cells in respiration. Exhaled air contains more carbon dioxide and more water vapor compared with inhaled air, and that's because these two substances are products of respiration carried out by our cells. So these substances get transported to the lungs so that they can be breathed out. Why does our breathing rate increase during exercise? I'm sure you're aware if you've gone on a run, you'll start breathing far faster, far more deeply. The reason for that is because your muscle cells in particular need to respire more. They need to release more energy to allow your muscles to contract. Respiration, remember, produces carbon dioxide. There are obviously lots of benefits associated with exercise on the cardiovascular system. It includes increased numbers of red blood cells, increase in the size and strength of the heart, decrease in resting heart rate, increased muscle mass and bone density, increased number of alveoli, and a decrease in resting blood pressure. One of the most straightforward methods we can use to measure our heart rate is by taking our pulse rate. Remember, you can feel that pretty much in your wrist and in your neck, although you might want to attach yourself to a monitor in order to get a more accurate representation of your pulse rate. The simplest way to see the effect of exercise on the heart rate is basically to take your pulse rate at rest for 1 minute, then do some exercise, and then measure your pulse rate directly afterwards for a further minute. Remember, the advantage of an increased heart rate during exercise is because the heart is responsible for pumping blood around your body. That blood contains the all-important glucose and oxygen needed by cells to carry out the respiration required to release energy and to enable our muscles to contract. You find that the fitter the person, the more they exercise, the lower their pulse rate, their heart rate tends to be. Now we'll talk about the effect of smoking on the body. So smoking is obviously really bad for you. There's lots of reasons for this. First of all, cigarettes contain tar. Tar is a carcinogen. This is a chemical which causes cancer. So people who smoke for a very long time may see increased likelihood of getting lung cancer. The other problem with smoking is that it paralyzes those cilia which line your trachea. Remember that these are cells which waft backwards and forwards, removing bacteria-laden mucus from our trachea so that it can be destroyed by the hydrochloric acid in our stomach. When our cilia is paralyzed, that bacteria stays in our trachea. Therefore, you see an increase in infection and an increased incidence of diseases like bronchitis. Long-term smoking may also cause the disease emphysema. Now, emphysema damages your alveolar walls. therefore effectively reducing their surface area, meaning that less gas exchange takes place. So a person really struggles to breathe out when they have emphysema, which is a chronic disease that can't really be treated. You'll often see people using oxygen tanks if they have emphysema. Cigarettes contain nicotine. It's an addictive drug. Nicotine actually increases the viscosity of the blood. It makes it thicker, meaning that you have an increased chance of high blood pressure and the coronary heart disease associated with that. Cigarette smoke contains carbon monoxide. Carbon monoxide, remember, combines irreversibly with the hemoglobin in red blood cells. This prevents the transport of oxygen, so it is highly toxic. A scientist investigates the effect of exercise on breathing rate. She measures the breathing rate in breaths per minute of 2 people P and Q every 5 minutes for 30 minutes. This is her method. Measure their breathing rate every 5 minutes while they exercise for 20 minutes. Measure their breathing rate every 5 minutes for a further 10 minutes while they recover from exercise. The table shows her results. Plot a line graph of the results for persons P and Q. Use a ruler to join your points with straight lines. Okay, I thought this would be a great opportunity in this video to show you how to draw graphs accurately. So the first thing to be aware of is what goes on the y-axis and what goes on the x-axis. What you measure, so your dependent variable, must always go on your y-axis. You must familiarize yourself with recognizing which of your data is your dependent variable, and then what changes your independent variable goes on your x-axis. Then it's just a matter of labeling your axes. Now you've worked out which way round they go, and do make sure you include units. So there's time in minutes. It's very important you get these the right way round. Breathing rate, which was breaths per minute. Don't reinvent the wheel here, just copy out the table headings. So yeah, there's your y-axis, here's your x-axis. Now we need to choose a sensible scale, occupying as much of the graph paper as possible. So in time in minutes, we go from 0 to 30. So try and space out these numbers. Yeah, I'm using a large portion of my graph paper. What about breaths per minute? What's our lowest value? 12. Highest value is 25. I'm going to go up in 5s on my y-axis. I need a key because I've got 2 people, so I'm going to start with person P. Make sure you plot those points really accurately and follow the instructions. It's asked us to join the points with straight lines. Now I'm getting ready to draw my second graph line for person Q. If you can hear that noise, which I'm sure you can, that is seagulls. I'm sorry, there's no getting away from them where I live. I've drawn both sets of data then for person P and person Q. I've joined my points with straight lines. You can choose not to do a key. If you're just going to use pencil in the exam, which I do recommend because you can rub out your points if you accidentally misplot one, just label each of the line, point out which one's P and which one's Q. That's fine. So if we're asked to explain the change in breathing rate during exercise, just checking the question, it says they exercise for 20 minutes. So let's make sure we're looking at the right portion of the graph. So I'm just looking at this portion of the graph. So I need to ask myself, why does the breathing rate increase during exercise? And there is a mark for pointing that out. So why would our body want to take more breaths so that we can get more oxygen into our body? And why do we need that oxygen? So that we can carry out more aerobic respiration. And what is the purpose of aerobic respiration? It's to release energy, and we use that energy to contract our muscles when we exercise. Explain why the breathing rates of persons P and Q remain high for 5 minutes after they have finished exercising. So why do they want to continue getting more oxygen into their bodies? Well, that's to remove any lactic acid that's built up as a result of anaerobic respiration, and it's to repay the oxygen debt. The time taken to recover from exercise is often a good measure of fitness. The scientists concluded that person P is much fitter than person Q. Comment on the validity of this conclusion. So when they want you to comment, You to say if you agree or disagree, and you need to make a statement regarding the investigation, so how you think it was carried out. So is person P fitter than person Q? So person P is the purple line. So the first thing we can see is that person P has a lower breathing rate at rest compared with person Q, and a low breathing rate at rest tends to be indicative of a fit person. We can also see that person P's breathing rate drops faster after exercise. Again, that could be indicative of them being fitter. However, these results don't automatically mean that person P is fitter, because actually if you look, they both return to their normal breathing rates in the same amount of time. In terms of commenting on the experimental design, we have no information about the age or the sex of the person. We've got no data about the exercise they actually carried out. And finally, the data is not reliable because they didn't repeat their experiment. Moving on to the transport topic now. So we're going to start by considering why unicellular organisms don't require a transport system. So here's a unicellular organism. It's made up of one cell, and it is actually an amoeba, which, remember, I taught you is an animal-like protoctist. Now, the amoeba doesn't require a transport system. And the reason for that is, being unicellular, it has a very large surface area to volume ratio. So there's surface area to volume ratio, meaning the diffusion distance is short. So what do we mean by that? If you were to calculate the volume of the amoeba, calculate its surface area, you'd find that it has a very large surface area to volume ratio. And what that in effect means is that no part of the amoeba is awfully far from the outside environment, meaning that diffusion of the oxygen, of the nutrients it requires, can occur really, really quickly. Whereas multicellular organisms, e.g. us, humans, they're made up of lots of cells. They have a comparatively much smaller surface area to volume ratio, meaning that diffusion occurs too slowly. And as a result, they require transport systems to deliver oxygen, etc., far more efficiently. I just want to quickly show you how to calculate surface area to volume ratio. So John's teacher suggested that the different size cubes have a different surface area and volume. Use the formulae below to calculate the surface area of each cube, the volume of each cube, and their surface area to volume ratios. So to find the surface area, we do 6 multiplied by the cube side squared. So we need to do 1 squared multiplied by 6. 2 squared, which is 4, multiplied by 6, which is 24. The volume we've been told is the cube side cubed. So do 1 cubed, which is 1. 2 cubed, which is 8. And then the surface area to volume ratio, that means you do the surface area divided by the volume. That's how you calculate that. So 6 divided by 1 is 6. 24 divided by 8 is 3. So plant transport now. 2 very important tissues. The first one I want to mention is the xylem. Now the xylem transports water and mineral ions. Do make sure you've learned that. We've talked about those mineral ions before. Nitrates, magnesium ions, required by the plant to keep it healthy. Crucially, it occurs in an upwards direction only. That makes sense because the water and mineral ions enter at the root hair cells. They cross the root, they enter the xylem, and then they move upwards to supply the leaves with the mineral ions and water it requires. In terms of its structure, it's highly specialized. It's worth learning that it's made up of dead cells which join end to end to form a continuous column. You can think of it like a drain pipe, which is going to help the movement of mineral ions and water within the plant. A further adaptation is lignin, which is a tough substance which further supports the xylem and gives it strength. The second tissue you need to know about is the phloem. This time the phloem transports sucrose, which is a type of sugar, as well as amino acids. And if you're feeling fancy, you can say that it transports them from source, where they're made, that's what the source refers to here, which will be the leaf, to a sink, where they're stored or used. Totally depends on the requirements of the plant. That could be the roots, for example. And this process of the movement of sucrose and amino acids in the phloem from source to sink is known as translocation. Because it totally depends on the requirements of the plant, the movement of sucrose and amino acids can be both up and down the shoot. And if we look at the structure of the phloem, it's not made up of dead cells. It's made up of sieve tube elements, which are these bits here, with sieve plates between them, which contain a small amount of cytoplasm. And then accompanying each of these sieve tubes is these special cells known as companion cells, which contain an awful lot of mitochondria. Remember, the role of the mitochondria is to release energy, so that energy is used to move the sucrose and amino acids into and out of the phloem. If we compare the xylem now, as I said, it's made up of dead cells which join end to end to form a continuous column. The movement of water and mineral ions occurs in an upwards direction only, and lignin reinforces the walls to give further support. If we look at the arrangement of these tissues within the plant, here's a stem. The outer layer is the phloem. This inner layer here is the xylem, and here we can see a microscope slide showing the arrangement of the phloem and the xylem. In the root, the setup is slightly different. Conveniently, the X represents the xylem, and then in between the prongs of the X, you find the phloem. We've mentioned the fact that the mineral ions are transported in the xylem, but now let's go into greater detail how they enter the plant. So I said that they enter via the root hair cell. Because they're moving from an area of low concentration to an area of high concentration, this movement occurs by active transport and it requires energy from ATP. However, water, because it's moving from an area of high water potential in the soil to an area of low water potential inside the root hair cell, water will move by osmosis across a partially permeable membrane. Transpiration now transpiration refers to the loss of water vapor from the leaf, and it occurs through small pores in the leaf known as stomata. If we think about the entire passage of water throughout the plant, we know that it enters the root hair cell by osmosis across a partially permeable membrane. The water flows into the xylem, and then it moves up the xylem to the leaf, where it then exits the leaf via the stomata. Now, the movement of water within the xylem is known as transpiration stream, and it occurs because the water molecules stick to each other due to hydrogen bonds which exist between them. This is something you'll study if you do higher level chemistry. The water molecules also stick to the walls of the xylem, creating a continuous column, and so as transpiration occurs at the stomata, more water is drawn up from below due to the fact that these water molecules are stuck to each other. Now, this transpiration stream has many benefits for the plant. It helps to cool the plant, it helps to transport water and mineral ions to the leaves, and remember that water is used in photosynthesis. It also helps to keep the plant turgid. So going back to transpiration, which is the loss of water from the surface of the leaf via the stomata, what factors increase the rate of transpiration? So the factors which affect transpiration include light intensity, temperature, wind speed, and humidity. You need to be able to describe and explain these factors. So in order to increase the rate of transpiration, you can increase the wind speed. And what that does is it blows water vapor away from the surface of the leaf, meaning that the concentration gradient, so that's the difference between the water levels inside the leaf versus the surrounding air, it means that the concentration gradient is steepened and therefore water will diffuse out of the leaf faster. So transpiration occurs more quickly. Now, in terms of humidity, you want dry conditions to increase the rate of transpiration, and that's again because you increase the concentration gradient between the leaf and surrounding air, so water will diffuse out more quickly. If you increase the temperature, the water molecules gain kinetic energy, and as we know, increased kinetic energy means that those water molecules will diffuse faster. Last up, light intensity. Now, when it's light, the plant wants to photosynthesize, so it will choose to open more of its stomata in order to allow the carbon dioxide into the leaf that it requires. As a result of those stomata opening, water is able to diffuse out of the leaf, so increased light intensity increases the rate of transpiration. If they ask you about factors which decrease the rate of transpiration, you need to say the opposite, so low light levels, high humidity, low wind speed and low temperature, and be prepared to explain them. Now, a potometer measures the rate of transpiration. So you have your cut shoot placed in the potometer, and as the leaf transpires, more water is drawn in at the cut shoot, and as a result, the air bubble you see will move to the left, and you can measure the distance moved by the air bubble in a particular time frame to find the rate of transpiration. This obviously assumes that the rate of water uptake into the shoot is the same as the rate of transpiration. Now, they like to ask you a lot about the experimental setup here. So the first thing you need to be aware of is that you need to cut the shoot underwater, and that's to prevent any air bubbles getting inside the xylem. Next up, if the leaves get splashed with water, you need to make sure you dry them because you don't want to create conditions of humidity if that's not the factor that you're investigating. You also need to introduce an air bubble into the potometer because we need to be able to monitor how far that air bubble moves in a particular time frame. Use Vaseline or petroleum jelly to seal any joints in the potometer, again to avoid any air bubbles. Transpiration is the evaporation of water from the surface of a plant. Explain why transpiration is useful for plants. So this is really a question asking why transpiration stream is so helpful. Transpiration stream helps to cool the plant and deliver water and mineral ions to the leaves. A student uses this apparatus to measure the rate of transpiration in still air and the rate of transpiration in windy conditions. The student calculates the total loss of water after 1 hour in milligrams per centimetre squared of leaf area times 10 to the minus 4 for the plant in still air. He then repeats this calculation for the plant in windy conditions. The graph shows the student's results. The total surface area from which transpiration occurs is 250 centimetres squared. Calculate the loss of water from this plant in still air in milligrams per hour. So make sure you're looking at the right bar. Read off the y-axis, we can see that 21.5 is the water loss after 1 hour in milligrams per centimetre squared of leaf area times 10 to the minus 4. So we need to include that times 10 to the minus 4 there. So 21.5 times 10 to the minus 4 equals 0.00215. But because we want it for the total leaf surface area, we need to multiply that number by 250. Explain the results shown by the graph. So why does more transpiration occur when it's windy compared with when you've got still air? We know that wind blows water away from the surface of the leaf. Steepening the concentration gradient between the inside of the leaf and the outside air, and as a result, diffusion of water out of the leaf occurs more quickly. Justify 2 factors that the student needs to control to make a valid comparison of transpiration in still air and in windy conditions. So remember, the factors which affect the rate of transpiration include light intensity, wind, temperature, humidity. So obviously these are all factors which need controlling in order to make a valid comparison, but they're asking us to explain it. So I'm going to say the same temperature needs to be used, as increased temperature, remember, increases the kinetic energy of the water molecules, meaning that they'll diffuse more quickly out of the leaf. And then it's up to you which one you pick. I'm going to go with light intensity because remember that affects the opening and closure of stomata. And obviously transpiration can only occur if the stomata is open. Focusing in on the blood. So what are the components of the blood? Well, you have both white blood cells and red blood cells. Remember, white blood cells consist of both lymphocytes and phagocytes. Red blood cells, their official name is erythrocytes. Remember, they're needed to transport oxygen. You've also got a jelly-like substance known as plasma and small fragments of cells called platelets. And we'll talk lots about their various roles a bit later on in this video. The blood is responsible for transporting oxygen from the lungs to respiring cells, carbon dioxide from respiring cells to the lungs, various nutrients such as glucose and amino acids, hormones, urea, which is a breakdown product of proteins from the liver to the kidneys where it's excreted. Kind of repeating ourselves a little bit, but just talking about the role of the plasma now. So the plasma, the jelly-like substance, transports hormones from where they're made glands to the organs that they act upon, their target organs, nutrients such as glucose from the small intestine to cells, urea from the liver to the kidneys, carbon dioxide from respiring cells to the lungs. So what I've just mentioned there is specifically the role of plasma, whereas before I was talking more generally about the role of blood as a whole. Focusing in on red blood cells, so as I've already said, red blood cells are responsible for transporting oxygen around the body. That oxygen binds to a pigment known as hemoglobin present on the red blood cells. The red blood cells have lots of adaptations to increase how much oxygen they transport. They have a characteristic biconcave dish shape. This increases their surface area to volume ratio, maximizing the amount of oxygen that can be transported. They have no nucleus. This provides more space for hemoglobin. They're flexible, which enables them to squeeze through blood capillaries. And they have a thin membrane, which provides a short diffusion distance. Have you ever wondered why athletes like to train at high altitude? It's because of the following. At high altitude, you find there's less oxygen present. To compensate, more red blood cells are produced by our body. More red blood cells means that there's more hemoglobin, meaning that more oxygen can be transported by our blood. More oxygen is great for athletes because it means that more respiration can take place, more energy released, and more muscle contraction. The white blood cells form the body's defense against pathogens. Remember, pathogens are microorganisms which cause disease. They include things like bacteria, fungi, protists, and viruses. So there are 2 white blood cells. The first type is the phagocyte, the second type is the lymphocyte. Phagocytes have a characteristic lobe nucleus. Phagocytes carry out phagocytosis. What this means is that they engulf the pathogen. They do this through that lobe nucleus. It means that they can engulf that pathogen into their cytoplasm, whereupon digestive enzymes are released to destroy the pathogen. The lymphocytes, these have a large single lobe nucleus. This is because they don't engulf pathogens. Instead, they recognize small proteins on the pathogen surface known as antigens. The lymphocytes secrete small proteins known as antibodies which bind to the antigen. Now, the lymphocytes, when they secrete these antibodies, first of all, the antibodies can help kill the pathogen, the bacteria. How do they do that? They can help cause the bacterial cells to clump together, making them more likely to be spotted by phagocytes so that they can then be engulfed. They also produce antitoxins, which counteract any toxins produced by the pathogen. And another method by which they may destroy the pathogen is by causing them to burst open. What about those small fragments of cells known as platelets? So To simplify their role, we would say that platelets clot the blood at the site of a wound. So as soon as you cut yourself, you don't bleed indefinitely. And the reason for that is because your platelets create a scab at the site of a wound. Real importance of all of this is to ensure that you don't have excess blood loss and that pathogen entry is prevented. If you want a more active approach to avoid getting ill in the first place, you might choose to have a vaccine. A vaccine is an injection containing a dead or weakened or attenuated form of the pathogen. What happens when that pathogen enters the body, it doesn't make you sick because it's dead or weakened, but your antibodies respond to the antigens on the pathogen. The lymphocytes produce memory cells so that upon re-entry of that pathogen, antibodies can be made again much sooner, much faster, and in much greater quantity. So vaccines are an excellent way of protecting yourself against a particular disease. Herd immunity is all about giving a large number of the population a particular vaccine. This reduces the number of potential hosts that can be infected by a pathogen, reducing the chance of this pathogen being transmitted. There's far less chance of a non-vaccinated person coming in contact with the particular pathogen when herd immunity has been used effectively. Human populations are at risk from infectious disease. Describe the different types of pathogen, refer to a disease that each type of pathogen causes in your answer. So obviously you need to know your definition of a pathogen, which is a microorganism which causes disease. So that includes viruses, bacteria, prototysts, fungi. 6 marks. So viruses are non-living organisms, e.g. HIV, which causes AIDS. So there's viruses done. Bacteria are unicellular, e.g. pneumococcus, which causes pneumonia. So you do need to know the variety of living organisms topic extremely well to answer this question. Prototysts, e.g. Plasmodium, cause malaria. Fungi, they just want a tiny detail to do with their structure, which have a cell wall made of chitin, may cause athlete's foot. Explain how vaccination protects humans from pathogens. Vaccines contain a dead or weakened form of the pathogen. They cause some lymphocytes to turn into memory cells. Upon re-entry of the pathogen, antibodies are produced faster, sooner, and in greater quantity. This is known as the secondary immune response. Now we turn our attention to discuss various blood vessels, starting with the arteries. You need to be able to describe their structure and function. So if we start with their structure, we say that arteries have a narrow lumen. This means that the hole that the blood flows through is very narrow. That's the lumen. Because of that, blood flows at high pressure, and to stop the blood vessels bursting, they need thick muscle and elastic fiber walls to withstand that pressure. Arteries carry blood away from the heart. Arteries tend to be oxygenated. The exception here is the pulmonary artery, which takes blood from the heart back to the lungs. That is deoxygenated. Veins have a much wider lumen. Because of this, blood flow is much slower at much lower pressure, and therefore they have thinner walls made of muscle and elastic fibers. Because that blood flows at such low pressure, sometimes it has a tendency to back up, and that's why it's a valve which ensures that blood flows in the correct direction. Veins tend to be deoxygenated. They contain no oxygen. The exception here is the pulmonary vein, which transports oxygenated blood from the lungs to the heart, specifically the left atrium. The capillaries now, these are much smaller than arteries and veins. They're only 1 cell thick. They have thin walls to provide a short diffusion distance. Their walls have small pores that allows the movement of substances. Capillaries have a very large surface area to volume ratio. Pulmonary means relating to the lungs. Renal means relating to the kidneys. Hepatic means relating to the liver. Use these words to help you label the various blood vessels around the body. If the main artery coming from the heart is known as the aorta, it feeds out of the left ventricle. Any vessel coming off the aorta therefore must be an artery. So the name of the vessel supplying the liver is the hepatic artery that supplies oxygenated blood to the liver. The vessel supplying oxygenated blood to the kidneys is the renal artery. Now if we turn our attention to blood vessels flowing away from organs, because these vessels will join the vena cava, they must be veins. Let's take the vessel leaving the liver, it must be the hepatic vein. The vessel leaving the kidneys must be the renal vein. Don't forget that the hepatic portal vein exists between the intestines and the liver. This will contain the highest concentration of glucose because it's coming from our gut. Do think about various other components of blood coming into and out of organs. It makes sense that blood going to the lungs must have the highest concentration of carbon dioxide, the lowest concentration of oxygen. Blood leaving the kidneys, remember the kidneys are an organ of excretion, therefore blood in the renal vein must be lowest in urea as that's been excreted in our urine. The heart is a muscular organ which helps to pump blood around the body. It's very important that it can do that because of what I've said previously. The blood transports a large number of very useful substances, the most useful of which is oxygen needed by cellular respiration. So in terms of the anatomy of the heart, it has 4 chambers, 2 atrium and 2 ventricles. Just be very careful when it comes to labelling the heart. The left and right sides are flipped compared with what you'd probably think, and that's because if you picked up the diagram and popped it into your chest, that's how the heart would actually appear. So notice that you have the left atrium, the left ventricle, the right atrium and the right ventricle. The separation between the left and right side of the heart, it's called the septum. A septum simply means a wall of muscle, such as your septum in your nose here. It keeps the left and right hand sides of the heart totally separate, and the reason for that is it stops the mixing of oxygenated and deoxygenated blood. The left side of the heart contains oxygenated blood. That means blood containing oxygen. The right hand side contains deoxygenated blood. That means blood containing no oxygen. So that's your broad layout of the heart. Next up you have vessels which supply the heart. Any vessel that comes into the heart is known as a vein. Any vessel which leaves the heart is known as an artery. Arteries carry blood away from the heart. Try and remember that those 2 A's come together. Right, let's get into the nitty-gritty of the heart then. So we're going to start with the left atrium. I've already told you that the left side of the heart is oxygenated. So where must that blood have come from? Well, oxygenation, gas exchange, occurs at the lungs. So blood supplied to the left atrium comes from the lungs, and the name of this vessel is the pulmonary vein. The left atrium contracts, forcing blood into the left ventricle. The left ventricle contracts, forcing blood into the main artery known as the aorta. This artery goes off and splits into smaller arteries supplying various organs and cells with oxygenated blood. The cells use up that oxygen in their respiration, so we say that the blood becomes deoxygenated. It obviously needs to return to the lungs in order to become oxygenated, but in order to do that, it needs to go to the heart first of all. It does this by travelling in the vena cava vein to the right atrium. The right atrium contracts, forcing blood into the right ventricle. The right ventricle contracts, forcing blood into the pulmonary artery. This blood is then returned to the lungs, where it can pick up more oxygen. There are various valves to prevent the backflow of blood in the heart. Between the walls of the left atrium and the left ventricle, you'll find the bicuspid valves, the left atrioventricular valves. They're both names for the same thing. And then in the walls between the right atrium and the right ventricle, you'll find the tricuspid valve, the right atrioventricular valves. These prevent backflow of blood back into the atrium. At the base of the pulmonary artery in the aorta, you have what's known as semilunar valves. These valves prevent backflow of blood into the ventricles. Remember, it's important that the left and the right hand side of the hearts are separate in order to prevent oxygenated and deoxygenated blood from mixing, as this would result in lower rates of respiration. We say that the human heart is a double circulatory system. The reason for this is that blood enters the heart twice for every one circulation around the body. If you look more closely at the anatomy of the heart, you'll notice that the wall of the left ventricle is thicker than the wall of the right ventricle. Why is this? Well, that thickness means that there's more muscle. And the reason why the left ventricle needs more muscle is it needs to generate a higher pressure to force blood much further around the body. Whereas remember, the right ventricle is simply pumping to the lungs. Next up, the coronary arteries. Now, the heart is the hardest working muscle in the body. It contracts myogenically all by itself throughout your lifetime. Now, the heart needs a huge amount of oxygen in order to carry out those contractions, and that's the role of the coronary arteries, which snake over the outside of the heart. The coronary arteries supply the heart with its own oxygen requirements. Now, what happens if you lead a fairly unhealthy lifestyle? If you eat too much saturated fat, potentially smoking, it can be caused by stress. It can even be hereditary, so caused by genetic factors. But what may occur is that fatty deposits may build up in the lining of the coronary arteries. This narrows the lumen of the artery, meaning that less oxygenated blood reaches the heart muscle cells. As a result of this, aerobic respiration can no longer occur. More anaerobic respiration occurs, leading to the buildup of lactic acid, and fundamentally the heart muscle cells die. The diagram shows a section through a human heart with the chambers labelled P, Q, R and S, which chambers contain deoxygenated blood, so we need the right side of the heart, which remember is this side, so it's P, which is the right atrium, and S, which is the right ventricle. So the answer here is B. Explain why two of the valves in this heart are closed. So the valves we're talking about are here in the base of the aorta and the pulmonary artery. So the valves are closed to prevent the backflow of blood. And say into which chamber? To prevent the backflow of blood into the ventricles. This ensures blood flows to the... Make sure you point out where the aorta and the pulmonary arteries supply blood to. So flows to the body, that's the role of the aorta, and lungs, which is the role of the pulmonary artery. Heart disease is a major risk to health in the United Kingdom. In a study, the number of people with heart disease was recorded. The table shows the results of the study. Give two conclusions that can be made from the study. First of all, we can see that males are more at risk than females from coronary heart disease. And again, if you look more closely at those figures, you can see that the risk of coronary heart disease increases with age. For both males and females. The population of the UK is 65 million, half of which are male, so that's 32.5 million males. Calculate the number of males with heart disease in the age range 18 to 44. So you have 5 people per 1,000 suffering from coronary heart disease. So we need to do 32.5 million as a number, divided by 1,000 and then multiplied by the 5 as given in the table to get 162,500. Explain how heart disease can affect a person's health. So coronary heart disease leads to fatty deposits in the coronary artery, meaning less oxygen is supplied to heart cells, less aerobic respiration occurs, so less energy released. Person is unable to exercise due to that lack of energy and may suffer a heart attack. Osmoregulation is all about controlling our blood water levels. The super important organ involved with osmoregulation is the kidney, but do remember that the kidney has 2 roles. It controls blood water levels, but it also excretes the substance urea. Remember, excretion is the removal of metabolic waste from our bodies. Substances which are excreted include sweat from the skin, carbon dioxide from the lungs, and then focusing in on the kidney, urea from the kidney, and that urea is excreted in urine. You have two kidneys. They're two fist-sized organs found in the lower portion of your back. They have various vessels which feed into and out of them. The blood vessel supplying oxygenated blood to the kidneys is known as the renal artery. The blood vessel removing deoxygenated blood from the kidneys is known as the renal vein. In terms of the layers of the kidney, the outermost layer is known as the cortex, the middle portion is known as the medulla, and then you have what's known as the pelvis. Now, the kidney contains millions of tiny structures known as the nephron, and it's important that you know the structure of the nephron inside out. The blood vessel which enters the nephron is known as the glomerulus. It's important that you're aware that the structure of the glomerulus is such that the blood vessel coming into it is wider than the blood vessel leaving, and that has important implications when we consider pressure. The first part of the nephron is known as the Bowman's capsule, and then you enter the first coiled tube, which unfortunately you need to be able to describe its full name of proximal convoluted tubule. We move into the loop of Henle or loop of Henle. Then we move into the second coiled tube, known as the distal convoluted tubule, before entering the collecting duct. That feeds into the ureter, which supplies the bladder with urine. Now, at the glomerulus Bowman's capsule boundary, a process known as ultrafiltration occurs. So ultrafiltration really means small molecules such as water, ions, urea and glucose are moved from the glomerulus into the Bowman's capsule under pressure. Now that pressure comes from the fact that the blood vessel entering the glomerulus is wider than the one leaving, generating pressure to force those small molecules out of the blood. Do note that things like proteins are too large to enter the Bowman's capsule and they remain in the blood. And if there's any protein present in your urine, then that can indicate damage to either the glomerulus or the Bowman's capsule, because it means the pressure is so great that those proteins have entered the nephron when ordinarily they would stay in the blood. The next process you need to be aware of is known as selective reabsorption. So if you consider the contents of the nephron, it contains ions, urea, glucose, water. Obviously, it's important for our body that we retain as much glucose as possible. Otherwise, what was the point of us eating food containing glucose? The point of our digestive system trying to absorb that glucose through the wall of the small intestine is because we want that glucose in our blood. But at this point, it's entered the proximal convoluted tubule. So selective reabsorption is really taking back that glucose and some ions back into the blood. It occurs at the proximal convoluted tubule, and crucially it occurs by active transport, and that's because the glucose moves from an area of low concentration to an area of high concentration. Therefore, there's the requirement of energy from ATP. So what that means is that the urea is allowed to stay in the nephron where it joins the ureter, is added to the urine, and at that point can be excreted from the body. So selective reabsorption and ultrafiltration are really important in the excretion of urea. What about the osmoregulatory role of the kidney? So we're going to take two scenarios, one when we've had too little to drink and one when we've had a lot to drink. So what happens when we have too little to drink? Well, obviously we want to retain as much water as possible in the blood, and the following steps occur in order to do this. So first of all, we have osmoreceptors present in the hypothalamus in our brain. These detect the low water levels of our blood. They send a signal to the pituitary gland to release more of the hormone ADH. ADH stands for antidiuretic hormone. The ADH travels in the blood to the nephron, and specifically it acts on the walls of the collecting duct, making them more permeable to water. This means that more water is reabsorbed into the blood. Less water is available to make urine, so resulting urine is concentrated, yellow in colour, low in volume. But what about when we've had lots to drink? Well, we're going to just learn those same steps but use slightly different wording. This time our osmoreceptors in the hypothalamus detect high water levels in the blood. The pituitary gland therefore secretes less ADH. The ADH travels to the collecting duct in the nephron. It makes the walls of the collecting duct less permeable to water, meaning that less water is reabsorbed, meaning that resulting urine is high in volume and dilute and light in colour.
The human kidney is an organ that is able to process excretory products from human metabolism. Concentration of urine is calculated using this formula. The concentration in milliosmoles per decimeter cubed equals the amount of waste divided by volume in decimeters cubed. The amount of waste the kidneys produce per day is 600 milliosmoles. The maximum concentration that the human kidneys can produce is 1400 milliosmoles per decimeter cubed, Calculate the minimum volume of urine that must be produced each day. Give your answer in centimetres cubed. I know this looks nasty, but it's actually okay. We just need to sub in all our values in the right place and do a unit conversion. So our concentration was 1400. The amount of waste was 600, and we're after the volume. So in order to work out x, we do 600 divided by 1400. That's in decimeters cubed. They want the answer in centimeters cubed, so make sure you're aware that to do that conversion, you need to multiply your answer by 1000. Name another organ that carries out excretion in the human body. I'm going to go with the lungs. Explain the role of the nephron in osmoregulation. So that's control of blood water levels. It's up to you which way you argue this. I'm going to say if blood water concentration decreases, the osmoreceptors in the hypothalamus detect this change. Remember, what they do next is they send a signal to the pituitary gland to release lots of ADH. ADH travels in the blood to the collecting duct of the nephron, increasing the permeability of its walls. This means that more water is reabsorbed into the blood, so resulting urine is concentrated and low in volume.
Homeostasis refers to the maintenance of a steady internal environment. So in humans, the types of conditions we try to keep constant include our temperature, because 37 degrees is the optimum temperature for enzyme-controlled reactions. We like to control our blood water levels as well as our blood glucose levels through the action of insulin and the hormone glucagon. Now, the coordination response topic includes lots of content regarding plants. So obviously plants need to respond to various stimuli, and a stimuli is a change in the external environment. Plants will respond to things like gravity, water, and light levels. And they do this through the redistribution of plant signal molecules known as auxins. They're not hormones, because remember, our definition of a hormone is that they're a chemical messenger carried in the blood. So for that reason, auxins aren't plant hormones, but you can think of them in a very similar way. So a plant's response to light is known as a phototropism, and parts of the plant carry out positive phototropism, which means they will grow towards the light. Some parts of the plant show a negative phototropism, which means they grow away from the light. So if we think about the particular organs of the plant which are responding in this way, shoots will obviously grow towards the light because they want the leaves to receive as much light as possible so that they can photosynthesize and produce glucose. Roots will show negative phototropism. They'll grow away from light because they want to grow downwards in order to obtain water and mineral ions and to help anchor the plant. But how does the plant grow towards the light? What happens is those auxins become concentrated on the side furthest from the light. Here they cause cell elongation, and it's the cells elongating which actually causes that stem to bend towards the light. A plant's response to gravity is known as a geotropism, and roots show positive geotropism. They grow down in the direction of gravity for the reasons we've already discussed. Shoots show negative geotropism. They grow away from the force of gravity. We can investigate tropisms through the use of coleoptiles, which are simple wheat seedlings. And Darwin is famous for carrying out an array of experiments which enabled him to study the effect of various stimuli on the growth of coleoptiles. Just to be aware, a clinostat is a useful piece of apparatus to be aware of. A clinostat is often used as a control in an experiment because it can negate the effects of gravity, water, and light.
I just want to show you a page from my revision guide. So just to show you how the auxin redistribution causes the shoot to bend towards the light. Here we've got light coming from one direction. The auxin molecules are made in the tip of the shoot and they diffuse to the side furthest from the light. They cause cell elongation on the side furthest from the light, meaning that the shoot bends towards the light. Here's a series of experiments involving coleoptiles, so those wheat seedlings, examining the action of auxin. So here we have light coming from one direction, and as we would expect, because nothing's happened to the plant shoot, the auxins are able to concentrate on the side furthest from the light, causing the shoot to bend towards the light. This time, we're cutting off the shoot tip and we're throwing it away, and look, as a result we see no bending of the shoot, and that proves that auxins are made in the shoot tip, because by cutting off the shoot tip we've removed the auxins, so we've removed the ability of the shoot to react to the light. This time we're placing an opaque cover over the tip. The auxins are unable to detect the light, and as a result we see no bending of the shoot. Here we've got an impermeable mica barrier inserted on the illuminated side. Remember, the auxins will diffuse to the side furthest from the light, so they're able to move as usual, and as a result we see the positive phototropic response of the stem. This time the mica sheet has been inserted on the shaded side, which is where you'd expect the auxins to diffuse to, and as a result they can't move properly, so we see no tropic response. And then lastly, rather than a mica block, we've got a permeable gelatin block that allows the auxins to diffuse, so it doesn't matter that gelatin block covers the whole of the shoot, the auxins are still able to diffuse to the side furthest from the light, so we see a positive phototropic response. Flowering plants can respond to their environment through tropic responses. These involve root or shoot tips growing towards or away from a stimulus. A student set up an experiment to investigate how shoots respond to gravity. He placed a plotted plant on its side and looked at its growth after 5 days. The diagram shows what he saw. Explain the benefit to the plant of this response. So despite the fact that the student's interested in gravity, we can see that the shoot has grown towards the light, so it's grown away from gravity, and the advantage of that is to obtain that light for photosynthesis. Give one variable that the student should keep the same in his experiment and suggest how he could do this. So if they're interested in investigating gravity, what they want to do is make sure the light levels are the same elsewhere. So light intensity should be the same everywhere. Suggest a suitable control for this experiment. We need an experiment where gravity won't be having an effect on the growth of the plant. So the best way of doing that is using a clinostat which continuously rotates the plant, negating the effect of gravity. Plants can respond to drought by closing their stomata. Suggest how a plant benefits from closing its stomata when less water is available in the soil. That will prevent wilting because it will stop transpiration. Explain the disadvantages to the plant of closing its stomata. No CO2 can enter if the stomata are closed, so less photosynthesis occurs and less glucose is made.
Now I want to talk to you about coordination and the nervous system. So the function of neurons is to transmit electrical impulses. It's important you're aware of the 3 different neurons, the motor neuron, the sensory neuron, and the relay neuron. Be familiar with the shape of these neurons. Notice that motor neuron has that characteristic star-shaped cell body. The relay neuron sits between the sensory and motor neurons, so it is symmetrical at each end. Neurons have long axons in order to transmit the electrical impulse. Be aware of which direction that impulse passes along the neuron. The axon transmits electrical impulses away from the cell body. It has a very narrow region of cytoplasm surrounded by a cell membrane, and the axon itself is a single elongated nerve fibre. The cell body is where you'll find the nucleus located. Remember, that controls the activity of the cell. Also present on the neurons are things called dendrons. These are short extensions which come from the cell body. The dendrons divide into smaller branches known as dendrites, and the dendrons are responsible for transmitting the electrical impulse towards the cell body. The axon has a fatty layer surrounding it known as the myelin sheath. As with all fat, the fat acts as insulation, and in this case helps to speed up the rate of the electrical impulse. If we zoom in on the sensory neuron transmits electrical impulses to either a relay neuron, a motor neuron, or the brain. The sensory neuron has a single axon which carries electrical impulses away from the cell body, and one dendron which transmits the electrical impulse to the cell body. The relay neuron sits between the sensory and motor neuron. It has short dendrons The motor neuron transmits electrical impulses to an effector, which is either a muscle or gland. The muscle responds by contracting, the gland responds by secreting a hormone. Motor neurons have a long axon and short dendrites. The human nervous system consists of the central nervous system. This is made up of the brain and spinal cord. The role of the nervous system is in the control and regulation of bodily functions. It can both be under involuntary and voluntary control by the brain. Responses can be split up into both reflex responses as well as a regular response. In a reflex response or a reflex arc, there is no conscious input from the brain. These are things which happen extremely fast, and the purpose of a reflex action is to protect us from harm, such as the time if you put your hand in a hot oven, you'll know that your hand withdraws itself without your need to register, oh, it's hot, oh, I don't want to burn my hand, oh, I must remember it. No. By that point, you'll have already removed your hand from the oven. So reflex actions are fast, they protect us from harm, and they don't involve conscious input from the brain. In order for our bodies to respond, we need something to respond to, and this is what's known as a stimulus. A stimulus is a change in the environment, so it could be a change in terms of whether you feel pain, whether there's heat, whether you're detecting something being hard or soft, whether you hear something, whether you see something. A receptor is a specialized type of cell which detects that change in environment. It detects the stimulus, so that could be your receptors in your eyes, your rods and your cones found in your retina, which are sensitive to light. Light is the stimulus in this case. There are lots of different receptors in your body which are sensitive to various different stimuli, such as light, pressure, temperature, taste, and pain. So if we think about what happens in a regular response, such as when our eyes detect light, say the rods detect low-level light, those are the receptors, then they're going to send an electrical impulse along the sensory neuron to your central nervous system, which is your brain in this case. Then an electrical impulse is sent along your motor neuron to an effector. In this case, it will be a muscle in your eye, probably the ciliary muscle, which will either contract or relax based on how far away an object is from your eye. In a reflex action, the steps are slightly different. So let's talk about the reflex action involved when you step on a sharp pin. So the stimulus is the pain. The receptors are present in your foot. An electrical impulse is sent along the sensory neuron to a relay neuron located in the central nervous system. That impulse passes to a motor neuron and then finally an effector. In this case, that will be a muscle which responds by contracting in order to remove your foot from that painful pin. Do remember that between two neurons is a gap. This gap is called a synapse. It's important that you're aware that at a synapse, there's no electrical impulse. Instead, a neurotransmitter is released from the presynaptic membrane. This neurotransmitter diffuses across that synapse and binds to postsynaptic receptors. Remember that electrical impulses can only pass in one direction across the synapse.
The diagram shows a human reflex response to touching a hot object. Some of the structures in the reflex arc are labelled. Let's have a look at what's going on here. So you can see that the receptor will be in the finger. That's going to be sensitive to the hot object. That's going to send an electrical impulse along the sensory neuron F to the reflex neuron G. X will be a motor neuron, which is attached to an effector, K, which will be our muscle which contracts to pull our finger away from the hot object. Give the name of this reflex response. You've got several options here. You can say it's an unconscious response, you can say it's involuntary. So that's quite unusually worded, but as long as you know the definition of a reflex action, you should be fine. Draw an arrow on the diagram to show the direction of the nerve impulse at point X. It's going to be moving in this direction. Which structure detects the stimulus? That's going to be E. Which structure is the motor neuron? It's coming along here. So make sure you pick an option that's provided so you can see that's I. A neuron is 1.1 meters in length. The speed of the nerve impulse is 120 meters per second. Calculate the time in seconds for the impulse to pass along the neuron. Give your answer in standard form. So you need this formula triangle, which states that time is distance divided by speed. Distance is 1.1 meters. Our speed is 120. And then double check the wording of the question. We need that in standard form. So it's 9.2 times 10 to the minus 3. Describe how the impulse passes from neuron to neuron in the reflex arc via a synapse. A neurotransmitter diffuses across the gap.
Now let's discuss the eye. So remember, the eye is a receptor organ. It is sensitive to light and it's adapted in order to create a clearest image as possible. So if we go through the various parts of the eye, you've got your conjunctiva, which protects the eye. You've got the outer protective layer known as the sclera. You have your cornea, which is at the very front of your eye. This refracts light as it enters the eye. That light passes through the aqueous humor and then through the pupil. So the pupil's role is to control how much light enters the eye, and it's muscles in the iris that will control the size of the pupil. The muscles in the iris are both the circular and radial muscles. We'll talk a lot about that shortly, but like I've already said, the iris controls the size of the pupil. Moving backwards in the eye, we reach the lens. The lens carries out the fine focusing of the light. It refracts the light. Now the thickness of the lens alters how much that light is refracted, and the ciliary muscle and suspensory ligaments will alter the thickness of the lens. Then you have what's known as the vitreous humor. This is a pressurized liquid that helps maintain the shape of your eye. The all-important retina is located towards the back of the eye. It contains photoreceptors called rods and cones, which are sensitive to light. The rods are sensitive to low-level light. The cones are sensitive to color. You have a blind spot, which is where the optic nerve leaves the eye. No photoreceptors are found here. The fovea contains a high concentration of cones. You'll have the choroid, which is a black layer that stops light reflecting around the eye. Make sure you're clear that it's the lens which is responsible for focusing on objects which are different distances away from the eye. So you need to learn the steps as to what occurs if you're looking at an object close to and far away. So starting with scenario 1, looking at an object close to, here the ciliary muscles contract, meaning that the suspensory ligaments slacken and your lens is fat. This means that the light rays are refracted more strongly to produce a clear image on the retina. If you look at a far away object now, this time your ciliary muscles relax, meaning that your suspensory ligaments are pulled tight. This has the knock-on effect of making your lens thinner and light is refracted less strongly. Now we'll look at what happens in your eye when you're in different light intensities. So what happens if you enter a very bright room? Well, you'll want less light to enter the eye because you don't want damage being caused to the retina. So how do you make less light enter the eye? Well, you need to constrict or narrow your pupil. The way in which that occurs is that the circular muscles contract, the radial muscles relax, and your pupil constricts. If you enter a room with very dim light, this time you want your pupil to be nice and dilated, nice and wide to allow as much light as possible to enter the eye. So this time your radial muscles contract and your circular muscles relax.
The diagram shows the difference in the pupil diameter of the eye in bright light and after moving into dim light. The difference is caused by reflex action. The table gives descriptions of parts of the reflex arc involved with the reflex action. Complete the table by naming each part. One has been done for you. Contains light receptor cells, that is the retina. Neuron that sends impulses into the brain, that is the sensory neuron. The microscopic gap between neurons is the synapse. Contains muscle effector cells, the iris. Explain how the lens changes when you view a near object. So the fact that you are viewing a near object means that the lens becomes fatter in order to refract the light more strongly. Now we need to decide what happens to cause that lens to become more convex. So the ciliary muscle contracts. And the suspensory ligaments slacken.
Homeostasis means the maintenance of a steady internal environment. Such internal environments include body temperature, glucose concentration of the blood as well as water levels of the blood. It's important that our body temperature remains fairly constant because if it rises too high, enzymes may be denatured, killing cells. If it's too low, then enzyme-controlled reactions occur too slowly. The skin is super important in thermoregulation, that means the control of our body temperature. But how does the skin help us control our temperature? How does it carry out thermoregulation? Let's think about when we're too cold. When we're too cold, our hairs on our skin stand up on end, trapping a layer of insulating air close to the skin, meaning that we're kept warmer in this way. Other mechanisms involved include shivering when our muscles contract, releasing heat. Vasoconstriction occurs in the arterioles in our skin. Remember, vasoconstriction means the narrowing of these arterioles, meaning that less blood flows close to the skin, less heat is radiated. Sorry about the seagulls. What about when we're too hot? This time you'll see vasodilation occur. This is when the arterioles dilate, more blood flows close to our skin, more heat is radiated. Our hairs on our skin lay flat, less insulating air is trapped. Sweating occurs. As sweat evaporates, it helps to cool the body. The layer of the skin are as follows. You've got the epidermis, which is the top layer, the dermis, and then the subcutaneous fat, which sits underneath this layer. Thermoregulation is a good example of negative feedback. This means that when a change occurs to the environment, mechanisms are brought about within our body to oppose that change.
So how do we control our blood sugar levels? So if our blood glucose levels increase, what changes occur in our body to reduce the blood glucose levels? So first of all, insulin is secreted by the pancreas. It travels in the blood to the liver, and what it causes to happen is the soluble glucose to be converted into insoluble glycogen. And that insoluble glycogen is stored in the liver. And remember, I taught you previously that the reason we like to convert our carbohydrate to an insoluble form is because when it's insoluble, it won't have any osmotic effect, which means it won't draw water into and out of cells. What happens if we've been asleep or if we've run a long race and our blood glucose levels have decreased? We'll want to increase them. So this time the hormone glucagon is secreted by the pancreas. And glucagon has the opposite effect of insulin. The skin is an organ of homeostasis and excretion. Describe what is meant by the term excretion. It is the removal of metabolic waste. Learn these definitions nice and accurately. That is worth 2 marks. The diagram shows a section through the skin with 2 structures labelled A and B. The structures labelled A and B play a part in homeostasis when a person enters a warm environment. Explain the role of structure A. So structure A is a sweat gland. So obviously its role is to release sweat, which cools the body when it evaporates. Explain the role of structure B. So structure B is an arteriole. So when we're very hot, remember the arterioles vasodilate, meaning more blood flows close to the skin, so more heat is radiated. Some hormones are involved in homeostasis. Explain the role of insulin in homeostasis. Insulin lowers blood glucose by converting soluble glucose to insoluble glycogen.
Now we move on to reproduction. So first of all, we need to start with a sperm and an egg cell. The sperm cell is what we call the male gamete. It is the male sex cell. The egg cell is the female gamete, the female sex cell. Remember that sperm are made in the testes under the control of the hormone testosterone. Eggs are made in the ovaries. Several hormones are important here: follicle stimulating hormone, luteinizing hormone, estrogen and progesterone all play a role. The moment that egg is released from the ovary, it then travels along the fallopian tube where it waits to be fertilized. The sperm enter the female's body during copulation. The sperm are deposited in the vagina. The sperm swims through the cervix, up through the uterus, where it fuses with the egg in the fallopian tube. This is the process of fertilization. Now, do remember that sperm and egg cells are both haploid. They contain one set of chromosomes each. The reason for that is that when they fuse at fertilization, that the first cell formed, the zygote, contains a diploid set of chromosomes. It contains two sets of chromosomes. So ensuring that the sperm and the egg have half the number means that the diploid number is restored when that zygote is first formed. In order to form our embryo, the zygote must then divide. The type of cell division responsible for that is called mitosis. So we'll go from 2 cells to 4 cells to 8 cells, 16 cells, and so on. Meiosis is a different type of cell division. Meiosis is used to produce the sperm and the egg cells. So meiosis takes place in the ovary and the testes. Meiosis is essential to this process because it introduces variation. It ensures that every sperm and every egg is genetically different, and that's important when we consider the offspring produced. Survival of the fittest states that there will be variation within a population, and those organisms better adapted are more likely to survive. So meiosis is one way of introducing variation. The other way is through random fertilization. You do not know which egg is going to fuse with which sperm. And this is why sexual reproduction has an advantage over asexual reproduction because of the variation produced. It helps in a changing environment. With asexual reproduction, when there's only the need of one parent, all offspring produced are genetically identical to each other and their parent, their clones. Although this is a super fast reproduction method, if conditions do change, it does mean that it's likely that all the organisms will die because none are better adapted to a particular environment. Let's focus in on great detail as to the structure of the male and female reproductive system. So starting with the male reproductive system, I've already mentioned you have the testes. These are responsible for sperm production, and it's under the influence of the hormone testosterone. There are two testes and they're held in a sac of skin known as the scrotum. The purpose of the scrotum is to, in effect, hold those testes outside of the body. This helps to lower the temperature, and it's that lower temperature which is vital for healthy sperm development. When that sperm is released, it passes along the sperm duct and various fluids are added from the prostate gland as well as the seminal vesicles. These provide nutrients to help the sperm to swim. The sperm then passes down a tube called the urethra. This will help convey that sperm as well as urine out of the male body. So the urethra is a tube which passes along the entire length of the penis, and it helps to convey sperm and urine out of the male body. The penis, as I've already said, it allows urine to exit the body. It also, in effect, places sperm within the vagina of the woman during copulation. Now we move on to the female reproductive system. So we'll start with the ovaries. The ovaries make the eggs. Remember, ovulation occurs, which is the release of an egg from the ovary, which is under the control of the hormone LH, luteinizing hormone. The eggs enter the fallopian tube. The other name for the fallopian tube is the oviduct. There are small cilia here. These are small hairs which help to move the egg down the fallopian tube where it sits and waits for any sperm. If copulation, sexual intercourse occurs while the egg is sitting in the fallopian tube, the person may become pregnant, the zygote, and then the embryo forms and the embryo implants in the wall of the uterus. So the uterus is where your fetus, your embryo, will grow for the 9 months, which is the gestation period for a human female. Gestation period just means how long she's pregnant for. The role of the cervix is to act as a boundary between the uterus and the vagina. It holds the growing fetus in place and forms the first part of the birth canal. The vagina is where sperm's deposited during sexual intercourse. It's also known as the birth canal because the baby will have to pass down the vagina in order to exit out of the body. Females also have a urethra, which is a tube used to convey urine out of the body. Now we'll talk a lot about the menstrual cycle now. So this is a cycle lasting approximately 28 days. It helps prepare the woman's body for a potential pregnancy. So we're going to be talking a lot here about hormones. So the first hormone you need to be aware of is FSH, follicle stimulating hormone. As the name suggests, it stimulates the follicles. These are the immature eggs found within the ovary. So in effect, FSH matures the egg. The next hormone that's important is known as LH, luteinizing hormone. This causes ovulation, the release of an egg from the ovary. In the meantime, you've got estrogen levels increasing. Remember, estrogen is produced by the ovary. Estrogen is responsible for thickening that uterus lining, ready for implantation. Progesterone is produced by both the ovary and the placenta later on in pregnancy. This helps maintain the thickness of the uterus lining. LH and FSH are made, remember, in the pituitary gland, so their source is actually the brain. As well as its impact on the uterus lining, estrogen also has a role of inhibiting FSH and stimulating the release of LH. Progesterone has the role of inhibiting the release of FSH and LH, and that makes sense. If you've already got an embryo developing in the uterus, the last thing you want is more eggs maturing and being released from the ovary. Now, what happens if no sperm enters the vagina? The uterus lining is no longer needed. It doesn't need to be thick anymore, so effectively it breaks down. It is shed, and this is what we call menstruation, and it's the blood loss which is associated with a period. So menstruation just means blood loss. It is the shedding of the uterus lining. Now, the placenta is a hugely important organ. It develops while the fetus is developing inside the uterus. It's an organ supplying the fetus with everything it needs for growth. So it will supply oxygen needed for respiration, glucose again needed for respiration. It will supply amino acids needed to develop muscles within that fetus, vitamins again needed for bone development, so that will be vitamin D as well as the calcium mineral ion. The placenta is also important for removing waste products produced by the fetus such as carbon dioxide and urea, and the placenta is adapted in order to ensure the transfer of these materials happens as efficiently as possible. It has villi which increase the surface area, meaning that diffusion of substances can occur much more quickly. It also has a good blood supply, lots of blood capillaries, and thin walls provide a short diffusion distance. The amniotic fluid is found within the amniotic sac, and it bathes the growing fetus, effectively acting as a shock absorber, meaning that the fetus is less vulnerable to any physical impact.
The diagram shows the human female reproductive system. I'm just going to practice labeling it. Make sure you can do this too when you're doing past paper questions. Here's your ovary, fallopian tube, uterus, vagina, cervix. Where does fertilization normally occur? That's within the fallopian tube, so that's A. State how estrogen and progesterone affect structure C during the menstrual cycle. So estrogen repairs the uterus lining, so C, remember, is the uterus, whereas progesterone maintains the uterus lining. In vitro fertilization, IVF, is used to help some people have children. Eggs are mixed with sperm in a laboratory. Embryos are then transferred into a uterus. Describe how an embryo forms after the eggs and sperm have mixed together. So the sperm fertilizes the egg to form a zygote. Mitosis occurs to increase the number of cells in order to form an embryo.
Sexual reproduction occurs in both animals and plants. Now, obviously in humans, reproduction is all to do with the male gamete, the sperm, fusing with the female gamete, the egg, in order to produce the zygote that through multiple cell divisions turns into an embryo, and later on a fetus and a baby. So that's the role of sexual reproduction in humans. In plants, it's slightly different. The male gamete is the pollen and the female gamete is the egg. So we need a way of getting the male gamete, the pollen, to meet with the female gamete, the egg. So how do plants do that? Well, they have two ways. You have a wind-pollinated flower and an insect-pollinated flower. With wind-pollinated flowers, clearly the wind blows the pollen onto another flower where it meets the egg. Fertilization can then occur. With insect-pollinated flowers, it's the role of an insect to transfer the pollen to another flower so that it can fuse with an egg. So as I've said, keep in mind the point of sexual reproduction in plants. So let's go through the ways in which the insect pollinated flower is adapted to encourage that insect to come and pollinate. First of all, it has large colourful flag-like petals that enables the insect to actually see the flower. They often have a strong scent that enables the insect to smell the flower. The pollen grains themselves tend to be sticky so that they actually stick to the legs of the pollinating bees. And remember, the insect doesn't carry out pollination because it's a kind, benevolent insect. It does it because the flower is going to offer it something, and that is in the form of a nectary. A nectary contains sweet nectar that the insect feeds upon. You'll find that the pollen is often enclosed in the petals, and that's to ensure the insect rubs against it on the way to the nectary, so it's actually guaranteed that it will pick up some pollen. With wind-pollinated flowers, you'll find that they have dull, small petals. After all, the wind doesn't care how beautiful the flower is. It doesn't need to see the flower. You'll find that there's no scent. You'll find that there's a lack of nectary. The pollen tends to be small and lightweight, so it can easily be carried by the wind. The anther, which contains the pollen, hangs outside so that the pollen can easily be blown away. And the stigma, which captures the pollen, will be feathery and have a large surface area in order to capture as much of the pollen as possible. So now let's go through all the steps involved in pollination and fertilization in a wind-pollinated flower. So first of all, we need pollination to occur. This is the transfer of pollen from the anther where it's made to the stigma. Remember, the anther and the filament form the male part of the flower. The stigma, style and ovary form the female part of the flower. So when that pollen transfers from an anther to a stigma, pollination occurs. The pollen lands on the stigma and a pollen tube grows down the style. This enables the pollen to be delivered to the ovary where it fuses with an egg, and that's the process of fertilization. The pollen actually enters the ovary via a special structure known as the micropyle. A couple of extra points. A structure known as the ovule becomes the seed, the ovule wall becomes the seed coat, and the ovary becomes the fruit. So we've made our seed. So the seed is laying dormant, waiting for its opportunity to germinate. It needs 3 conditions to germinate: water, oxygen, and warm temperatures. The water is needed to activate enzymes. The oxygen is needed for respiration. The warm temperatures are needed to provide an optimum temperature for the enzymes involved in germination. So what is germination? Well, what you'll see happen is the seed coat bursts, a small shoot known as a plumule grows upwards, and a small root known as a radicle grows downwards. The seed can't photosynthesize at this point because it has no leaves, so it has its own food store which it will use up initially. When the first few leaves start to grow, at this point photosynthesis can take place and the plant starts to produce its own glucose. Now, asexual reproduction in plants requires only one parent. It produces clones, which are genetically identical offspring. A good example of asexual reproducers are strawberry runners and potato tubers. Humans can also artificially carry out asexual reproduction by taking a cutting. That's when you use a pair of scissors to cut off a small piece of plant which you dip in rooting powder containing auxins. This encourages root formation. There's another type of asexual reproduction called micropropagation, and we'll talk about this a little later.
The diagram shows a wind-pollinated flower with some structures labelled W, X and Y. This is the stigma, here's your filament and the anther. Describe how structures W, X and Y are adapted for wind pollination. So W, the feathery stigma enables more pollen to be captured. Y, the long filament hangs outside the flower. X, exposed anther means pollen can easily be blown away. Structures W, X and Y are adapted for wind pollination. Give 2 other differences between wind-pollinated flowers and insect-pollinated flowers. Wind-pollinated flowers have no scent. Wind-pollinated flowers have no nectary.
Let's now consider DNA. So first of all, the structure of DNA. DNA is a double helix. That means it consists of two strands. The strands' backbones are made out of sugar and phosphates, and they're joined by complementary bases. Remember that adenine always binds to thymine and cytosine always binds to guanine. These bases are joined by hydrogen bonds. A nucleotide consists of three subunits. It's the phosphate, the sugar, and a base. Remember that the sugar in DNA is deoxyribose. DNA is a comparatively long molecule, especially when you compare it to RNA, which is much shorter. DNA replication occurs before mitosis. Remember that mitosis is a type of cell division. DNA replication simply means producing new DNA from old DNA. Now that new DNA will consist of one of the old strands from the original DNA molecule and one new strand which is formed from the original template. The DNA produced is genetically identical to the original parent DNA. Now we'll move on to RNA. Right, we've already said that RNA is a shorter molecule compared with DNA. The sugar in RNA is ribose, whereas in DNA it's deoxyribose. DNA, remember, was double-stranded. RNA is in fact single-stranded. The bases vary slightly in RNA. You still have adenine, you still have guanine and cytosine. The difference here is that thymine is replaced with uracil. Broadly speaking, the role of RNA is to take information from DNA in the nucleus to the ribosome, which fundamentally allows protein synthesis to take place. Moving on to mutations now. So mutation is a spontaneous, rare and random change in the DNA of a person. Certain chemicals may increase the likelihood of a mutation taking place, such as carcinogens, such as tar. Ionizing radiation, UV light, exposure to X-rays and gamma rays can all increase the incidence of mutation occurring. A mutation in DNA can lead to a change in the base sequence. This alters the amino acid sequence, meaning that a different protein is made, potentially with a new phenotype. The effect that the mutation has totally depends on the type of mutation. So if you're adding extra bases as part of your DNA mutation, that will clearly change the amino acid sequence in its entirety. Sometimes when you simply have a substitution of bases, you actually end up with the same amino acids being brought to the ribosome. We'll talk a lot more about that very shortly when we go into the detailed explanation as to how protein synthesis works. So protein synthesis is split into two stages, transcription and translation. Transcription takes place first of all inside the nucleus. Here the hydrogen bonds holding together those complementary bases break. The DNA unwinds, exposing its bases. There are free RNA nucleotides present in the nucleus which pair up with the exposed DNA bases. This enables a single-stranded mRNA molecule to be formed. That mRNA now leaves the nucleus and binds to the ribosomes in the cytoplasm. It's important that you're aware that three bases on mRNA is known as a codon. The codons are complementary to anticodons, which are found on one end of a tRNA molecule. At the opposite end of the tRNA is an amino acid. So effectively, the order of codons dictates the order in which the amino acids are brought to the ribosome. The amino acids join together to form a polypeptide, which folds into a complex 3D shape to form the end protein. Remember that in terms of the possible number of codons, the way in which you work that out is by contemplating the fact that 3 bases is responsible for each amino acid. So if you do 4 cubed, that tells you that there's a potential 64 codons.
The molecules RNA and DNA are both involved in inheritance and protein synthesis. Give two differences between the structure of a DNA molecule and the structure of an RNA molecule. DNA is double-stranded, RNA is single-stranded. DNA is a longer molecule. This sequence shows the order of bases on a DNA strand. Give the sequence of bases in the mRNA that would be produced from this strand. Because you're making mRNA, remember that uracil, replaces T. So it's worth just lining it up here. We have G pairing with C. If it was DNA, A would pair with T, but it's mRNA, so we'd have a U. And just carefully work your way along. Take your time here. You don't want to make any mistakes. And then rewrite that. GUA, GUA, GGA, GUA, GAU. The sequence of bases in the mRNA is used to code for the amino acids in a protein. Calculate the number of bases required to code for an amino acid chain of 1400 amino acids. So 3 bases equal 1 amino acid. So obviously the number of bases we need is 1400 times 3, which is 4200. A mutation results in a change in the sequence of bases in a DNA strand. Discuss what effect a change in the sequence of bases could have on the functioning of the enzyme produced. So a change in DNA bases could lead to new codons on mRNA, meaning a change to the amino acid order brought to the ribosome. The amino acid order obviously dictates the polypeptide, so the polypeptide and therefore the enzyme made will have altered structures. Think about how an enzyme functions. The substrate may no longer bind to the enzyme's active site. This is a really detailed answer. But remember, you're not guaranteed brand new codons. Because multiple bases can code for the same amino acids, sometimes there's actually no change in the amino acid sequence. However, substitution may lead to some codons coding for the same amino acid, and as a result, no change to the enzyme occurs. Give one way that the incidence of mutation could be increased: exposure to ionizing radiation like UV light.
We now turn our attention to the hereditary topics, so genetics. We need to start by going through a whole list of various definitions, and it is important that you know the meaning of all these terms so you can apply these things to the questions when you're asked. So starting with an allele is a form of a gene, such as having blue eyes or brown eyes. They're both alleles for eye colour. They're given by letters, so blue eyes being a recessive trait will have a lowercase b denoting it. Brown eyes being a dominant trait will have a large uppercase B denoting it. A gene is a section of DNA which codes for a particular protein. The genotype is the alleles belonging to an organism. You'll have 2 alleles, so if you have brown eyes, for example, you could be uppercase B, uppercase B. Blue eyes would be lowercase b, but that is your genotype, whereas your phenotype is the physical appearance of a trait. So that is whether you have brown eyes or blue eyes. It is not the same as the genotype. Make sure you're clear on that, and we will look at some past paper questions to help you here. Homozygous means that you have 2 of the same alleles, such as 2 lowercase b's or 2 uppercase b's. Heterozygous means that you have 2 different alleles, such as a capital B and a lowercase b. A recessive trait, the alleles are only expressed in the homozygous state. That means you'd need lowercase b. As soon as that uppercase B is present, then the dominant trait would be expressed. Dominant is when only one copy of an allele is needed to express a particular trait. This is different to codominance, where both alleles are present in the phenotype. A good example here is the snapdragon plant. If you have a red snapdragon flower, a white snapdragon flower, rather than one colour being dominant over the other, what would happen is that both alleles would contribute to the phenotype, and in this case you'd see a pink flower. Moving on to haploid and diploid. Haploid means containing only one set of chromosomes. In humans this is 23 chromosomes. Diploid means containing 2 sets of chromosomes. In humans this is 46. Monohybrid inheritance means that one gene is responsible for a particular phenotype. Polygenic inheritance means that many genes are responsible for a phenotype, and in actuality that does tend to be the case for most traits. In most cases, many genes contribute to a particular phenotype.
So let's take a simple genetic cross. A heterozygous brown-eyed mother and homozygous blue-eyed father want a baby? What is the probability that their baby will have blue eyes? So we're going to use the following layout in order to make sure we answer this properly. Please don't rush this. You want to write mother, father, phenotype, genotype, gametes. So this is the preparation you do in order to make sure that you get your Punnett square nice and accurate. So set yourself up like this. You should have plenty of space in the exam to do this. Now look at the clues given in the question. We're going to use B to represent eye colour. So we know that we have a heterozygous brown-eyed mother. The fact that it's heterozygous means that the genotype must contain one uppercase B and one lowercase b. We know that the father is homozygous and blue-eyed. Blue, remember, is a recessive trait. Homozygous means that those alleles are the same. So there's the father's genotype. We're now ready to write down the answers in our table. So the phenotype of the mother is that she has brown eyes, because remember, phenotype is the physical appearance. The father has blue eyes. Now list the genotype. We know the mother, I've already worked it out, is capital B, lowercase b. Father is lowercase b, lowercase b. The gametes, this means the mother's sex cells, so this is the egg cells. Her egg cells will either have a big B allele or a lowercase b allele. The father's sperm are all going to be that lowercase allele. This means you're now ready to set up your Punnett square. So it should look something like this. Just list those alleles again. It's up to you if you circle them. Give yourself plenty of space. And the Punnett square will enable you to work out the possible likelihood of that baby having blue eyes. So just list the various genotypes. And you should give yourself space to list the phenotypes underneath. So this baby here will be brown-eyed. This one will be blue-eyed, brown-eyed, blue eyes. So in terms of answering the question, our probability is that there's a 0.5 chance of the baby having blue eyes. Although percentages aren't technically probability, you'd also get away in biology with writing 50% chance or 1 in 2 chance. But it's important that you do this because these questions tend to be worth around 4 marks, and that will enable you to get the marks for linking the various phenotypes and genotypes at the end. So I do implore that you spend the time writing this out properly. Let's take an example now involving cystic fibrosis. So we have a carrier mother and a carrier father of cystic fibrosis. Cystic fibrosis is a nasty disease caused by the recessive allele. I'm going to call that C. So in order to have cystic fibrosis, you need 2 lowercase c's. So the carrier mother automatically tells me that her genotype must look like this, the same for the father. It's because they contain that lowercase allele, which is responsible for causing cystic fibrosis, but because it's a recessive disease, they've got this healthy allele, which luckily for the carriers means they don't suffer with any of the effects. But if we actually have a look at the cross this time, we're going to use the exact same layout as before: mother, father, phenotype, genotype, gametes. Like I said, it works every time. So you can write healthy or carrier here, it's up to you. I've already worked out the genotypes. So half of those egg cells will be healthy, half will contain that lowercase c, same for the sperm. And then if we look at the Punnett square, that's a healthy child. Here is a carrier, still healthy though. It's that final child here which will have cystic fibrosis. We can say that these ones here are carriers and that this one's entirely healthy. So it's 25% chance or 0.25 chance of having cystic fibrosis.
I've already said that a diploid number of chromosomes means 23 pairs of chromosomes. One pair is actually known as the sex chromosomes, and this determines whether someone is male or female. Males have an XY sex chromosome combination, females have an XX chromosome combination, and it's possible to do a genetic cross to show this.
Let's now look at sex inheritance. So we know that the mother is going to be female, the father is male. I've already said that the sex chromosomes in females is XX, in males it's XY. So that means all of the female's eggs will contain an X. Half of the male sperm will be female, the other half will be male because they'll contain a Y. And again, you're ready to get going. So this baby here will be female, this baby here will be female, this baby here will be male, this baby here will be male, hence why there is a 50-50 chance of having a girl versus a boy.
Pedigree analysis is used to show how a particular genetic condition is passed on from generation to generation. They are in effect family trees. So you will have the grandparent level, the parent level, the grandchild level, and you'll be able to see a male and a female and the children they produce, and then using the key you'll understand whether they have a particular condition or not. The great thing about pedigree analysis or pedigree tree is it allows you to work out whether a particular condition is a recessive or dominant condition. So for example, if two healthy parents, and you'll be able to use the key to work out if they're healthy or not, have a child with the condition, it tells you that the condition is recessive and that both parents must have been carriers. That's the only thing that can be true if they're both healthy but their child has the condition. Equally, you'll be able to work out based on the number of people suffering from a particular disorder whether it's a dominant condition or not based on how many people have it in that pedigree tree. And we'll look at an example now to really take you through step by step so you can work out the sort of thinking you'll need to unpick these questions.
Familial hypercholesterolemia, FH, is an inherited condition caused by a dominant allele. People with the condition have high levels of cholesterol in their blood, and this increases the risk of dying from blocked arteries. The diagram shows the pattern of inheritance in several generations of a family with FH. So as I said, here's the all-important key down here that will tell you that all the squares are males, all the circles are females. If it's shaded in, then they must have the disease. If it's blank, then they are healthy. So the first thing you want to do is work out what genotypes would be required to cause FH. We know it's a dominant condition, which means that either this combination or this combination will cause the disease FH. The only way you can be healthy is to have the recessive genotype, which looks like this. It's very important that you get to grips with what the information is telling you. So if we look at part A, person A is heterozygous for FH. Use this information to complete the table. So we've been told that this person has two different alleles. We can immediately label all of the unaffected healthy people's genotypes because I've already said they must be lowercase h, lowercase h. So we're being asked how many people are homozygous recessive. Homozygous means they have two of the same allele. Recessive tells me that they're both lowercase. So then it's simply a matter of just counting up all of the homozygous recessive people. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. What about homozygous dominant? Homozygous, two of the same allele. Dominant tells me that they're both capital H. So now we need to work this out. This daughter here has the disease. We know that she'll have only got a lowercase h from her mum. What about this son over here? Same setup. He has the disease, so he must be uppercase. But he's got that lowercase h from his mum. And you just keep working it down. Look, it's the same combination here, so I can immediately write that allele combination, that genotype. Then working down to person N. They'll have inherited the lowercase h from their mum, the uppercase h from their dad in order to have the disease. The same is true for all three offspring. So actually, if you sit back and look, how many people have this genotype? Well, the answer here is 0. Person G and person H have 3 children who all have FH. What is the probability of G and H having 3 children who all have FH? So G and H are here. Let's do a super quick Punnett square to work out the answer to this. So these 2 people here will be diseased, so 0.5 chance of having the disease because they have this genotype. So in order to work out the chance of them all having FH, because it's probability in maths, remember if it's person 1 and person 2 and person 3, you need to multiply that probability 3 times to get 0.125 as your answer.
How did the huge variety of organisms which live on the Earth today come into being? Well, we use Darwin's theory of evolution and natural selection to explain this. So your definition for evolution is that all the organisms alive today, and many more which are now extinct, evolved from simple life forms which first came into being over 3.2 billion years ago. Natural selection explains how organisms became adapted to their environment. So let's take the giraffe as an example. Why did giraffes develop such long necks? So the first stage in describing the mechanism of natural selection is to say that there's variation within the giraffe population due to genetic mutation. Remember that a mutation is a random and spontaneous change in the DNA of an organism. And as a result, those mutations lead to some giraffes being better adapted to the environment. So, for example, some giraffes had longer necks which enabled them to reach more food. The longer necks enabled the giraffes to survive, and as a result of surviving, they could reproduce. By reproducing, they were able to pass on these favourable genes to their offspring, so their offspring also had longer necks, and the process repeats over several generations. And we can use this framework, which I'll add now, to describe natural selection in a whole variety of organisms. We can also use natural selection to explain why antibiotic resistance is becoming an issue. So antibiotics are used to kill bacterial diseases, and over the years they've been used very effectively. However, increasingly antibiotic resistance is becoming an issue whereby pathogens which were previously killed by the antibiotics are no longer affected. So how do we use natural selection to explain this? Well, again, there's variation within the bacterial population due to mutation, and some of the bacteria develop a mutation that enables them to not be killed by the antibiotic. Clearly, these bacteria then survive and they reproduce, and before long, the entire bacterial colony is now resistant to that antibiotic. The most famous example is MRSA, methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is a bacterium which was previously killed by methicillin, which is a relation to the more familiar penicillin. MRSA was a huge issue in hospitals a few years ago, and we are seeing an increased number of superbugs which are resistant to these antibiotics.
The diagram shows sandpipers feeding. Sandpipers have long beaks that they can dig for worms in the mud. Explain how sandpipers evolved to have long beaks. This is your bread and butter when it comes to a natural selection type question. It's always worth a large number of marks. So we're going to start by saying by the mechanism of natural selection. Now let's go through that scaffold I just talked about. There is variation within the sandpiper population due to mutation. Some evolved to have longer beaks, which enabled them to reach more food. These sandpipers were more likely to survive and reproduce and pass on their favourable genes to their offspring.
Ecology and the environment. To begin with, there's a whole bunch of annoying definitions you simply need to learn off by heart. So we'll start with the definition for ecosystem. This is the interaction between all the species and their environment. The definition of environment, the total non-biological components of an ecosystem, e.g. the water, the soil and the air. The habitat is simply the place where the organism lives. The population is specifically all the individuals belonging to the same species present in an ecosystem. Community, this is the population of all the species found within a particular ecosystem. They often ask you how to gain an estimate for the number of plants in a particular area. So for example, the number of daisies in a sports playing field. So how would we do that? Well, the method you need to describe involves the use of a quadrat, which is a wire metal frame, tends to be 1 meter by 1 meter. Because obviously we can't count the number of daisies on every single square inch of the field, we use a quadrat to get a sample. So one of the marks available is for saying that you place the quadrat randomly using a random number generator. What that means is if you break up the field into a grid, so for example, along one side of the grid you go A, B, C, D, on the other side of the grid you go 1, 2, 3, 4, etc. Each meter of the square will have a different grid reference, and you can use a random number generator to help you place that quadrat randomly within the field. You then count the number of plants present in your quadrat, you place several quadrats, and then you multiply up your answer to gain an estimate for the whole field. Biodiversity refers to the variety of animal and plant species present within an ecosystem. Biotic factors refer to living factors which affect a particular species. So for example, what kind of living factors affect the rabbit population? Well, that could be the presence of any pathogens which cause disease within the rabbit population. It could be the presence of predators like foxes which feed upon the rabbits. It could be the presence of other rabbits which compete for mates, food or breeding sites. Abiotic factors refers to non-living factors. If we're considering the abiotic factors which affect a plant, it could be the soil pH, it could be the availability of water, mineral ions, light intensity, carbon dioxide concentration. So do make sure you can list a variety of abiotic and biotic factors. Now, one part of this topic which comes up a lot is food chains and food webs. Food chains and food webs are very tightly interlinked. A food web is simply made up of multiple food chains. Now, a food chain represents the energy transfer within a particular ecosystem, and it always starts with a producer, which is an organism which photosynthesizes. It makes its own food. For example, grass uses sunlight energy to make glucose. To the right of the producer in a food chain, you'll place the primary consumer. This is the organism which feeds upon the producer, and the arrow always points to the primary consumer because that's the flow of energy. Then you have the secondary consumer, followed by the tertiary consumer. Sometimes you'll have a quaternary consumer, but most often you won't, and that's because the energy runs out higher up the food chain, meaning there's not enough energy to support a quaternary trophic level. So what is a trophic level? It's a stage of a food chain, such as the producer level, the primary consumer level. So why isn't there enough energy to support the quaternary consumer level? That's because huge amounts of energy are lost at each stage of the food chain, approximately 90%. So where do those energy losses occur? Well, if we take a food chain that involves a grass as the producer, followed by a primary consumer, a rabbit, and then a secondary consumer, a fox. So energy losses occur from the producer, the grass, to the rabbit, the primary consumer, because the rabbit doesn't eat all the grass. Some of the grass is indigestible. Energy losses occur from the primary consumer, the rabbit, to the secondary consumer, the fox. Think about why. Because some of the rabbit is inedible again. You can't eat its teeth. The fox won't be able to eat its fur. There are energy losses due to respiration, due to the rabbit carrying out excretion, so that's the removal of urine, digestion, so the removal of feces. It will also use up energy keeping itself warm and through its own movement. And that explains why some unethical intensive farming practices keep animals close together, for example, pigs kept in tiny pens, because it stops them moving around, it stops them getting cold, it helps conserve their energy so they can convert that to meat for human consumption. A pyramid of numbers shows the number of organisms at each trophic level, and it's irrespective of their biomass. So sometimes you find that pyramid of numbers aren't pyramidal in shape. For example, the bottom level always consists of the producer, and if you have a single oak tree, that's only one organism, so you'll have a narrower base compared with as you move up through the food chain, where you move to something like a sparrow. There'll be more sparrows than oak trees, so you'll see a slightly misshapen pyramid of numbers. Because biomass refers to the total amount of living material within an organism, you do find that pyramids of biomass are pyramidal in shape, because you'll find that the producer has greater mass compared with organisms further up the food chain.
I'm going to use this food web here to explain how food webs work, and then we'll answer the past paper question. So a food web always starts with the producer. This is an organism which photosynthesizes. It makes its own food, hence it's a producer. Don't let them confuse you in the exam. Look, this is the start of our food web, the diatoms. I know you probably don't know what that is. It's a single-celled algae, and this is actually going to be photosynthesizing. So yes, it's our producer. The next stage up from your producer is your primary consumer. It consumes foods, and you'll see the energy transfer always occur from the producer towards the primary consumer, hence why the arrowheads are pointing in this direction. So if we ask ourselves how many primary consumers we have, it's 2. Then moving up another stage, so you go from producer to primary consumer. Then you move on to the secondary consumer, and then the tertiary consumer. And as I said, the arrowheads will always show the direction of energy flow. Do be aware that some organisms occupy 2 trophic levels. For example, the mudworms here, if you follow this route, are primary consumers. If you follow this route, they are secondary consumers. So do be prepared for that. So let's answer this question. The diagram shows a food web. Use information from the food web to complete the table. The first one has been done for you. What is the number of organisms? 8. Double check. Yes, it's 1, 2, 3, 4, 5, 6, 7, 8. What is the number of producers? So how many organisms do we have photosynthesizing? It's just the diatoms, so that's 1. The number of primary consumers, so let's go up 1 trophic level. The zooplankton is a primary consumer, and the mudworms are a primary consumer if you go up in this direction. So the answer here is 2. What is the number of food chains? This is difficult. You're going to have to count how many different individual routes there are from the diatoms. So this is 1, 2, 3, 4, 5, 6. So we've dealt with that bit. 7, 8, 9. And then your 10th food chain is diatoms, mudworms, ghost crabs, sandpipers. That's tricky. Scientists collect data from a grassland ecosystem. For each trophic level, they determine the mean number of organisms in a square metre, the mean dry mass of these organisms in a square metre. The table shows the scientist's data. Draw a labelled pyramid of numbers for this data. So pyramid of numbers means we need to be looking down this column. Remember that the order will always go producer followed by primary consumer, secondary, and then tertiary, showing the flow of energy. So we're going to start with our producer at the bottom of our pyramid of numbers. It wants to be labelled, so there's my producer, and I had 592 individuals. Now my primary consumer. Secondary consumer. There was only 35 of those. And then lastly, the tertiary consumer. There was only 3 of them. Describe how you would collect data to find the mean number of producers per square metre in the ecosystem. So we're really counting the number of plants. The best way to do that is to use a quadrat. So place a quadrat randomly using a random number generator. Count all the producers present. Divide total sum of producers by area. The mass of organisms at each trophic level is called the biomass. The percentage of biomass in the producers that is transferred to the primary consumers is 4.5%. Calculate the percentage of biomass in the secondary consumers that is transferred to the tertiary consumers. So we're looking here So we've only got 2.4 grams in the tertiary consumers compared with 10.6 grams in the secondary consumers. We do 2.4 divided by 10.6 times by 100. So the percentage of biomass transferred is 22.6%. Comment on the energy transfers in this ecosystem. In your answer, refer to data from the table and the percentages of biomass transferred. So we need to talk about these numbers. So we can see that energy is lost at each trophic level. Let's compare. More energy is lost from the producer to the primary consumer compared with the secondary to tertiary consumer. And because it's worth 4 marks, it's worth saying where those energy losses occur. Energy losses occur due to excretion and digestion, respiration, and not all the organisms are eaten. And then commenting further on the data, there are fewer organisms, so we're talking about the pyramid of numbers now at the top of the pyramid, and that's again due to all those energy losses.
The carbon cycle describes how the element carbon is cycled through the atmosphere and the bodies of living organisms. For me, the best place to start is with carbon dioxide in the air. Green plants use the carbon dioxide in the process of photosynthesis to make glucose, which has the formula C6H12O6. So in this way, carbon is transferred. The green plants respire, which releases carbon dioxide into the atmosphere. The plants are eaten by animals and the carbon is transferred to their bodies. The animals respire and carbon dioxide is released into the atmosphere. So we can already see how readily carbon is moving between the atmosphere and living organisms. Eventually, the plants and animals die and they're decomposed by bacteria and fungi. The decomposers also respire, further releasing carbon dioxide to the atmosphere. The burning of fossil fuels in the process of complete combustion also releases carbon dioxide into the atmosphere, but obviously this is a human process. Some carbon gets locked up in fossils, which are found in sedimentary rock.
The diagram shows a cycle that is important for the recycling of biological molecules. Processes in the cycle are labelled A, B, C, and D. Give the name of this cycle. I can see straight away because the wood and fossil fuels are being burnt and they're releasing carbon dioxide. That's one stage of the carbon cycle. Which process reduces CO2 in the atmosphere? That's going to be photosynthesis because carbon dioxide is a reactant in photosynthesis. So just make sure the arrowheads are pointing in the correct direction. It's going to be A because that's taking CO2 from the atmosphere and using it to make the plant material. Name the 2 groups of microorganisms that carry out process B. So soil microorganisms, which are helping to break down dead material, they'll release CO2 through their respiration. So that includes bacteria and fungi. They're both types of decomposer.
The nitrogen cycle describes how the element nitrogen is moved between the atmosphere and living organisms. It's a bit more complicated compared with the carbon cycle. Personally, I prefer to start with nitrates. Now, nitrates are a type of mineral ion found in soils. Plants like to absorb lots of nitrates in order to build plant protein. How do they absorb the nitrates? Through the process of active transport, which occurs into their root hair cells. The nitrates move from an area of low concentration in the surrounding soil into the root hair cell, which contains a higher concentration. This therefore requires energy. The plants use the nitrogen to build plant protein. The plants are eaten by animals and the nitrogen is transferred. And we'll add some more detail to that in a minute or two. Now, eventually the plants and animals die and they are decomposed by decomposers, which include bacteria and fungi. Those decomposers produce ammonium ions. Nitrifying bacteria present in the soil carry out the process of nitrification. They take the ammonium ions and they convert them to firstly nitrites, followed by nitrates. This is a good process because it increases the fertility of the soil. Now, there are some annoying bacteria known as denitrifying bacteria which carry out the process of denitrification. Unfortunately, they take those mineral ions, the nitrate ions, and convert them to nitrogen gas, which is unreactive and therefore incredibly unhelpful for plants. Nitrogen-fixing bacteria are found freely in the soil as well as on the root nodules of legumes, which include peas and beans. These are fantastic bacteria because they take nitrogen gas and convert it directly to nitrates. This massively improves the fertility of the soil and helps to explain why farmers will sometimes choose crop rotation where they plant a legume every 4th or 5th year in order to improve the fertility of the soil. The Haber process is a process whereby humans take nitrogen gas and hydrogen gas and convert it to ammonia, which can then be used to make nitrogen-containing fertilizer. Now remember that once the nitrogen enters the animal body through the process of feeding, that's not the end of the story. The animal's digestive system will break down the protein using protease enzymes present in the stomach to amino acids so that they can be absorbed through the wall of the small intestine.
The diagram shows the nitrogen cycle with 4 different stages labelled A, B, C and D. Name the processes A, B, C and D. So we're looking at A initially, which we can see here and here. So these are bacteria found in the root nodules of legumes as well as freely in the soil. They're going to be carrying out nitrogen fixation. What about B? So now we're looking here. That's called decomposition. C, we're converting ammonium ions to nitrate ions. This is a good process. It's known as nitrification. And then finally D, this is a super annoying process. It removes those lovely nitrate ions and converts it to inert nitrogen gas. So this is the opposite process. It is denitrification. Name 2 different groups of organisms that can act as decomposers. That's fungi and bacteria. The nitrogen in a nitrate ion in the soil can become the nitrogen in a protein molecule in an animal. Explain how this happens. 4 marks. Right, the nitrate ions are absorbed into the root hair cell. Let's talk about the movement of these things. By active transport, the plant uses the nitrate ions to make protein. The animal, keep in mind the wording of the question, eats the plant and the nitrogen is transferred. And then what happens to the protein in the animal? Protein is digested by the enzyme protease. So it can be converted into soluble amino acids, which can then be absorbed through the wall of the small intestine and used to make animal protein. Farmers sometimes add fertilizer to the fields in which they grow their crops. Suggest 2 advantages of using animal waste, so that's animal feces, poo, as a fertilizer rather than using a chemical fertilizer. First of all, poo's always going to be cheaper. And secondly, there'll be less environmental pollution. If you're using something natural, it's actually less likely to cause eutrophication.
Human impact on the environment. So what kind of human activities adversely affect the environment? First of all, burning fossil fuels which contain sulfur impurities releases sulfur dioxide into the air. This can dissolve in rainwater to form acid rain. Secondly, the high temperatures found in car engines can cause nitrogen and oxygen in the air to react, forming nitrogen oxides. Again, these dissolve in rainwater, forming acid rain. Acid rain is a real problem because it lowers the pH of watercourses like streams and rivers, which adversely affects the aquatic organisms. Acid rain damages trees as well as limestone buildings. Now, if you study chemistry, you'll be aware of both complete and incomplete combustion. Complete combustion, when there's a plentiful supply of oxygen, produces carbon dioxide. Incomplete combustion occurs when there's insufficient oxygen present and carbon monoxide may be produced. This is a real issue because carbon monoxide is highly toxic. It combines irreversibly with the hemoglobin in our red blood cells, preventing them from transporting oxygen, so you may suffocate if you're exposed to too much carbon monoxide. The greenhouse effect is an important effect which increases the temperature of the Earth's atmosphere. The sun emits energy which enters the Earth's atmosphere. Energy is absorbed and re-emitted as longer wavelength infrared radiation. Some of the infrared radiation is radiated back into space. However, some of it is absorbed by certain greenhouse gases. This actually increases the temperature of the Earth's atmosphere. And if too much of this effect occurs, we say that global warming has taken place. So what are the examples and sources of greenhouse gases? Well, starting with methane, CH4, methane is produced by rice paddy fields as well as the digestive systems of cows. So effectively, when they burp and they fart, they release methane, which is a greenhouse gas. A second greenhouse gas is carbon dioxide, which, as we know, is released by burning fossil fuels. Other examples of greenhouse gases include water vapor, CFCs, which come from aerosols and old fridges, as well as nitrous oxide. Now, the real issues with the enhanced greenhouse effect come from global warming. When the temperatures rise, it causes the polar ice caps to melt. This releases a huge amount of water into the surrounding seas and oceans, meaning that their levels rise. Those rising sea levels flood low-lying land, leading to a loss of animal habitat and a destruction of biodiversity. Global warming is also responsible for the extreme weather, the large number of storms we've seen in recent years, as well as change in bird migration patterns. Now, burning fossil fuels, chopping down trees. So how can we reduce the buildup of greenhouse gases in our atmosphere? By planting trees, by burning fewer fossil fuels, by using alternative energy sources, renewable ones such as wind, solar and wave power, reduce cattle farming and the planting of rice paddy fields. Now we're going to move on to the pollution of rivers in a process known as eutrophication. So eutrophication is brought about when farmers add fertilizers to their soil. In heavy rainfall, these fertilizers may be washed into surrounding rivers. This is known as leaching. And the addition of fertilizers to the watercourses causes the buildup of algae. This is known as algal bloom. And the algae block the light for aquatic plants, meaning that they can't photosynthesize and they die. Large amount of dead plant matter provides food for bacteria. The bacteria decompose the plant matter, and as they do that, they carry out aerobic respiration, which uses up the oxygen within the river. Before long, there's no oxygen available for aquatic organisms, including fish, so these organisms die. Deforestation refers to the removal of trees. Why do we cut down trees? To provide space to build houses, to plant crops, to keep animals like cows and sheep. We also use the wood as a biofuel and as a building material and for paper making. Deforestation is a real issue for so many reasons. First of all, when you cut the trees down, there are no longer the roots available to hold down the topsoil. When it rains, therefore, surface runoff occurs, which means that all the nutrients and mineral ions present in the soil get washed into surrounding rivers, which may lead to eutrophication, but certainly means that the topsoil becomes infertile. When you cut down trees, it means that they're no longer alive to photosynthesize, meaning that they can't use up carbon dioxide in the air. Cutting down trees also releases carbon dioxide. When you cut down trees, there's a disturbance to the water cycle. Because the trees are no longer transpiring water, you won't get the same cloud formation, the same rainfall, and as a result, desertification may occur.
This passage describes water pollution caused by untreated human sewage and by fertilizer. Complete the passage. If sewage gets into fresh water, it will increase the number of pathogenic bacteria. Yep, because poop is full of bacteria. The sewage contains waste organic material in the form of, it's going to be feces, which is the scientific way of describing poop. Microorganisms break down this material using a process called aerobic. Respiration always follows aerobic. This process reduces the level of oxygen in the water, making it less likely for larger organisms to survive. Fertilizers can get into the water by a process called leaching. That's when the fertilizers wash into the surrounding watercourses. The minerals present in the fertilizer, such as nitrates, cause the rapid growth of plants in the water.
Food production. So why do farmers choose to grow lots of their crops inside polytunnels and greenhouses? Let's think about the various advantages. So the first advantage is that both greenhouses and polytunnels trap infrared radiation. I'm sure if you've ever been strawberry picking, you'll notice how much hotter it is within the polytunnel. This raised temperature is great for the plants because it optimizes the temperature for the enzymes involved in their chemical reactions, including photosynthesis, meaning that they can grow faster as a result of making glucose more quickly. The other good thing about polytunnels and greenhouses is they trap water released by transpiration. This increases the humidity of the polytunnel, therefore actually reducing transpiration rates, which again is good for plant growth. Now, in the winter, it's possible to artificially heat the polytunnels as well as to add artificial lighting so that it's light for 24 hours a day. They often grow tomatoes in this way in Iceland. And again, it means that the plant can grow all day long. You can also optimize the carbon dioxide concentrations within greenhouses and polytunnels. And remember, carbon dioxide concentration is a limiting factor of photosynthesis, so it is worth providing an optimum concentration. Farmers will also provide mineral ions such as nitrates because plants use them to build protein, as well as magnesium ions, which the plants use to make chlorophyll found in chloroplasts. Pests are frequently an issue for farmers, for example aphids, which damage the phloem of plants feeding directly on the sucrose present in the phloem. There are two approaches to killing these pests. One is a chemical pesticide. These are chemicals which the farmer applies directly to the crops. They tend to be very effective, they're very expensive, they're non-specific. It means that as well as the pest, they'll be killing lovely pollinators like honeybees, but they are easy to use. An alternative and more kind approach is using biological control. This is when you add a predator species which hunts and kills the particular pest. A good example is a farmer buying a whole load of ladybirds which they add to their crops which feed upon the aphids. Despite the fact that they are supposed to be specific and that, for example, the ladybird should be feeding on the aphids, it's hard to control what the ladybird is actually feeding upon. The other issue is that it can be a much slower way of killing the pest because it takes a while for the ladybirds to feed. But the great thing about this approach is it's a self-sustaining approach. Those ladybirds will reproduce, meaning that, technically speaking, this approach could work for many years. The other issue with pesticides is that bioaccumulation and biomagnification may occur, which is when that pesticide builds up within a food chain and becomes toxic to other organisms within the food chain.
Farmers sometimes use biological control to reduce the damage to their crops caused by pests such as insects. Which of these is an advantage of using biological control over chemical control? It lasts a short time. That's not an advantage, that would be a disadvantage. And the whole point of biological control is that it's self-sustaining, so it's not that. It leads to bioaccumulation, that's not a positive thing, so it's not that. It is specific, yes, that is an advantage of using biological control. It's quicker, it's definitely not because the animals take quite a while to eat the pests. Aphids are tiny insects that have very sharp mouthparts. They push these mouthparts into the phloem found in stems. They then feed on the phloem contents. Name two substances the aphids obtain from the phloem, that's amino acids and sucrose.
Yogurt making, a particularly niche part of the specification. If we add the bacteria Lactobacillus bulgaricus to milk, it will convert the lactose in that milk to lactic acid. They do this anaerobically, so they respire without the use of oxygen. In terms of the actual steps involved in yogurt making, first of all, you heat the milk to around 90 degrees. This is to sterilize the milk in order to kill any pathogens present. You then cool the milk to around 40 degrees to provide the optimum temperature for the lactobacillus bacteria. The bacteria then added and left for around 6 hours. This provides them with enough time to convert the lactose to lactic acid. If we're interested in monitoring how quickly they're carrying out this process, the best way to do this is look at the increased acidity of the mixture over a set time period. You can use a pH monitor to do this. So remember, the pH will lower over time due to the production of lactic acid.
Scientists carry out an experiment to see if reducing the availability of oxygen affects the production of yogurt. They use increasing acidity as a measure of yogurt production. They record the acidity of two cultures, one with reduced oxygen level, one with normal oxygen level, over 210 minutes. The table shows their results. Explain why increasing acidity can be used as a measure of yogurt production. So you need to think what happens in yogurt production is that the lactobacillus converts the lactose to lactic acid. Therefore, we have increased acidity. And that will get you your 2 marks. Give one abiotic variable that the scientists should control in their experiment. So we need a non-living factor, so temperature. Explain why the changes in percentage acidity are different in the reduced oxygen level and in the normal oxygen level cultures. So check out the data. You can see that the acidity has increased faster at reduced oxygen levels. So why does lower oxygen mean that the bacteria is working faster? That's because the bacteria carries out the all-important anaerobic respiration in reduced oxygen level conditions, meaning lactic acid is made quicker.
A fermenter is a vessel used to grow microorganisms on a large scale. Let's talk about the components of a fermenter. Depending on whether the organism carries out anaerobic or aerobic respiration, there may be an air inlet. This allows the oxygen in for aerobic respiration. Because the microorganisms release a lot of heat when they're respiring, you'll often find a temperature monitor, just to keep an eye on the temperature, as well as a cooling jacket, which will help reduce the temperature to prevent the denaturation of enzymes. A stirrer or paddle will constantly mix the contents of the fermenter, ensuring that the nutrients and heat are evenly distributed. It's important to sterilize the fermenter before you add your microorganisms. That's to kill any pathogenic bacteria present. The best way to sterilize is to use steam, because steam contains just water molecules. You would prefer not to use a disinfectant like Dettol because that will contaminate your end product. Remember, we can use a fermenter to make things like beer or to make things like hormones such as insulin used to treat type 1 diabetics. Another microorganism which is super useful is the one involved in beer making. It's the single-celled fungi yeast. Yeast takes glucose and converts it to carbon dioxide and ethanol. So the ethanol is used in beer making. The carbon dioxide can be used to help bread dough rise. Again, you'll need anaerobic conditions, ones which don't require oxygen. Fish farming. This is when fish are kept in tanks or enclosures. You get a lot of fish farms in lochs in Scotland. So the way fish farming works is they use nets in order to prevent predation from animals like seals and birds. They'll also use nets to separate fish of different sizes, and that's to stop larger fish eating the smaller fish. These fish are fed diets high in protein. Selective breeding programs are used to improve the quality of the fish, basically to make sure they're packing on as much flesh as possible. Aeration involves adding oxygen to the tanks so that the fish can aerobically respire. They're constantly cleaned to remove any uneaten food or feces to prevent the buildup of bacteria, which would use up the oxygen in their respiration and spread disease. Unfortunately, antibiotics are often given to the fish in order to stop bacterial disease. But remember, antibiotics can enter our food chains, which is why this isn't a great idea.
The diagram shows a salmon fish farm in the sea, suggests 3 ways in which the net protects the salmon. So let's have a look at what's going on. We've got our salmon and it has nets. We can see that the net will protect those salmons from predatory birds, this seal, as well as wild salmon, which won't eat the salmon, but it could compete for food. Waste food and feces can collect in the mud beneath the fish farm, suggests how this could affect the growth of the salmon. It's definitely going to reduce the growth. Why? Because the waste food and feces lead to the buildup of bacteria. And really this is a eutrophication style question. The bacteria use up the oxygen in aerobic respiration, which means there's less oxygen available for the fish to use for their own respiration. Suggest what should be done with the dead salmon with the fungus growing on it. Obviously, we want to remove dead salmon to stop the spread of pathogens, which, remember, are disease-causing microorganisms. Give the example of biological control shown in the diagram of the fish farm. It's right here. It's the wrasse which feeds upon the sea lice. So the wrasse is going to control the sea lice population.
Selective breeding is different from natural selection. Selective breeding is all about improving the quality of an animal or a plant to encourage favourable characteristics. For example, if you wanted to develop beef cows with an awful lot of muscle mass, you would select 2 cattle with the desirable features, so large muscle mass. You would cause that cow and that bull to breed together. You would select offspring that also show large muscle mass, and the process would repeat over several generations. Selective breeding is different from natural selection because in selective breeding, the human picks the animal and it's a much faster process.
Farmers have used selective breeding to produce cows that give high milk yields. Describe how selective breeding can be used to produce cattle that give high milk yields. So the farmer selects a high milk yield cow. The cow breeds with a bull. Offspring are selected, which also produce high milk yields. The process repeats over several generations.
Now, genetic modification or genetic engineering refers to the alteration of genes in microorganisms in order to produce a desired product. A good example of a desired product is the hormone insulin. Now, type 1 diabetics are unable to make insulin. Historically, pigs and cows were used, but there were ethical issues to do with their slaughter, and obviously the insulin is not perfectly suitable for humans and can lead to an immune response where that insulin is in effect rejected. So genetic engineering is a great way of making human-grade insulin, and I'm going to go through the following steps right now. So your first step in the genetic modification of bacteria is to use a restriction enzyme to cut open a bacterial plasmid. Your second step is to cut out the human insulin gene, in this case, using the same restriction enzyme. You then want to insert the human insulin gene into the bacterial plasmid using a second enzyme known as ligase. There are complementary sticky ends which are used to seal the human gene into the bacterial plasmid. The plasmid is then replaced back into the bacteria, and at this point, because the bacteria contains new genes, it's known as a recombinant bacteria. Do remember that the plasmid in this case is acting as a vector because it's allowing the transfer of biological material, in this case a human insulin gene. The bacteria are then placed into a fermenter where the conditions are perfect to ensure that they replicate as quickly as possible. We may also choose to genetically modify plants in order to give them desired characteristics such as a longer shelf life, resistance to frost. We may improve their nutritional value. For example, golden rice has been genetically modified to include vitamin A. This helps the people that consume the rice to see in the dark. Some modified tobacco plants produce hepatitis B antigens. This has potential use in the manufacture of vaccines. And some plants have been modified to make them more resistant to weed killers. Remember, a weed killer is a herbicide, and it's important that when you apply a weed killer that the crop plant itself isn't killed. So if you genetically modify that crop plant, it's less likely to be killed by the weed killer. Transgenic refers to the transfer of genes from one species to another.
Insulin is a hormone produced in the human body. Which organ produces insulin? It is the pancreas. Describe the role of insulin. It lowers blood glucose by converting glucose to glycogen. Some humans cannot produce insulin and need injections of insulin every day. This insulin can be obtained from cows, but this insulin might cause an immune response in the human body. Why does cow insulin cause an immune response in humans? So in this case, your cow insulin is going to act as an antigen which will be recognized by the lymphocytes in the body of humans, and it will be those lymphocytes that stage the immune response. So the answer here is B. The insulin now used for injections is obtained from bacteria that have been genetically modified. Describe how these bacteria are genetically modified to produce human insulin. So we know that we remove human insulin gene using a restriction enzyme. Insert the gene into the bacterial plasmid using the ligase enzyme. An industrial fermenter is used to grow the genetically modified bacteria. Explain why the fermenter is cleaned using steam before the genetically modified bacteria are added. So why do we need to clean it in the first place, and why do we use steam rather than a disinfectant? So we'll start by saying the steam kills pathogenic bacteria. Steam won't contaminate the insulin. The fermenter is sterilized.
Let's talk about cloning now. Now, cloning is producing genetically identical organisms. We could choose to clone a plant and we could choose to clone an animal, and I'll go through the different ways in which we can do that. So the most straightforward way of creating genetically identical plants is by taking a cutting. When you use a pair of scissors to cut off a small piece of plant, you dip it in rooting powder containing auxins and plant it in soil. This is a very slow way of creating a clone. A more technologically advanced method is using micropropagation, otherwise known as tissue culture. This occurs in a laboratory setting. The scientist uses a scalpel to obtain small pieces of plant known as explants. They place the explants on a sterile nutrient medium. It's sterile to reduce the chance of pathogen contamination. Auxins are provided, as well as humid conditions and the correct temperature and light intensity. Together, this ensures that the explants can grow nice and quickly, and micropropagation is a fantastic way of making a large number of genetically identical plants at any time of year. Moving on to the cloning of animals, the most famous cloning example was Dolly the sheep. She was the first animal to be cloned. But how do we clone an animal? Well, our first step is to obtain a body cell of the animal to be cloned. It's important that we use a body cell because we need the full complement of chromosomes. We need a diploid number that you find in a body cell. You then remove its nucleus, and we call this enucleation. You then need to obtain an egg cell from a female of the same species. You need to enucleate the egg cell, which means remove its nucleus. Your next stage is to insert the body cell nucleus into the empty egg cell. You're going to use an electric shock to fuse the two together. The cell then divides by mitosis in order to form an embryo. At this stage, you can place the embryo into the uterus of a female of the same species. We call this female a surrogate because she has no genetic relation to the embryo.
Many mammals have been cloned since Dolly the sheep was produced by cloning in 1996. It took 277 attempts at cloning to produce one sheep. Calculate how many attempts would have been needed to produce 50 cloned sheep in 1996. Okay, that's not too difficult. That's 50 times 277, so we would have needed 13,850 attempts. Describe the stages used to clone a male dog. So we need to remove a body cell from the male dog and enucleate it. Remove egg cell from female dog and enucleate. Place body cell nucleus into empty egg cell and use an electric shock to fuse. Cell divides by mitosis to form an embryo. embryo placed in a surrogate's uterus. Pet owners can now pay scientists to use cells from their pet to produce a clone. The process costs $50,000 for a dog and half of that for a cat. Cloning is not always successful, and as with Dolly, many attempts may be needed. Many scientists disagree with the use of cloning. Comment on whether cloning of pets is a good idea. Use info in this question, your own knowledge to support your answer. It's worth 4 marks. So we're commenting, so we need to say whether we agree or disagree whether the cloning of pets is a good idea. Cloning pets is good as it produces a genetically identical pet with similar features to the one that died. This may help stop the owner from grieving. Now let's point out the reasons why we think it's a bad idea. However, as you can see from the wording of the question, cloning is very expensive. Many attempts are required and it reduces genetic variation. Now we're going to tackle the CORMS style questions which come up in every single paper. Do make sure you've done plenty of practice here because these are worth 6 marks and you do absolutely need to be achieving every single one of those 6 marks. Now I personally don't like the CORMS layout, I prefer the variable layout, and you do need to know your variables anyway, so it shouldn't be any extra work for you. So when I'm asked to plan an investigation, I start by stating my independent variable, so that's what I will change. And we will go through an example so you can see exactly how I'm applying this scaffold to a question. The dependent variable is what I will measure. Control variables are what I will keep the same, and I suggest you provide at least 3. For reliability, you want to repeat and calculate an average, so you need to state that, and you usually get a mark for stating a time frame. Do make sure that time frame is sensible to the investigation being examined. So in the exam, I would personally remember this scaffold. I need to state my independent variable, my dependent variable, a minimum of 3 control variables. I'm going to repeat and calculate an average, and I need to specify a time frame. To show you how well this works, here's a past paper question. The diagram shows an insect called wasp. Wasps kill their prey by injecting a poison called venom through a small tube called a stinger. Some scientists believe that the smell of venom attracts other wasps. Design an investigation to find out if the smell of venom attracts other wasps. Include experimental details in your answer and write in full sentences. So yeah, this might seem a little overwhelming, but let's use my scaffold. Let's start with my independent variable. What will I change? I will use different concentrations of venom. Secondly, my dependent variable, what will I measure? So what am I measuring in this investigation? It's all to do with whether venom attracts other wasps, so I will count, make sure it's tangible, the number of wasps attracted. Here it makes sense to count the number. Never ever say I will measure the amount. Don't use the word amount, they don't like it. There's always a better word like number, mass, length, volume. These words are infinitely better than the word amount. Okay, my control variables, so what will I keep the same? I will keep the following the same. This is to ensure a fair test. So obviously we need the same species of wasp, and I'm even going to say and gender. Same volume of solutions. Think about other things which might impact wasp behaviour, potentially the weather, the time of year. So I'm giving lots and lots of control variables. I'm going to specify that I'm going to repeat and calculate an average, and then my time frame, let's pick a sensible time frame. I will run this experiment for 12 hours. So that's personally how I would go about answering this question. And just to show you how nicely that scaffold works, let's look at the mark scheme and see where we would have been awarded marks. Yes, I mentioned different concentrations of venom. Then I said I was going to count the number of wasps. In terms of my control variable, I stated that I'd use the same species and age. That's still the same mark, so I'll only give myself 1 mark there. I said I'd use the same volume of solutions, and I said I'd carry it out at the same time of year. I mentioned that I would repeat, and I mentioned a suitable time frame. So 1, 2, 3, 4, 5, 6, 7. I actually think I would have gotten 7 points there, so I would have definitely achieved my 6 marks.